welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner.
Well, welcome everyone to our FDA Clinical Investigator Training Course, 60 as we love to call it, our 2023 presentations. Good day, good afternoon, or good night, depending on where you may be. It is so exciting to see so many of you online with us here today. We certainly appreciate your commitment in attending this event and overcoming any scheduling problems or challenges that you may have had. We invite you to chat amongst yourselves in the chat pod when it is available, and we have wide global representation in the chat pod. What an encouragement to see such lively participation. My name is Brenda Stoddard, and I'm with the Small Business and Industry Assistance SBIA program, which operates within the Division of Drug Information, which in turn resides in FDA's Center for Drug Evaluation and Research's Office of Communications. It is my extreme pleasure, along with my colleagues, to be with you here today for this annual training course. It is a definite privilege to be able to collaborate once more with our Office of Medical Policy to bring you this two-day event specifically designed to promote professionalism in the clinical trial industry. We entreat you to make the most of this experience Attend all the sessions that interest you, learn all that you can, and submit your questions during the available Q&A sessions, and of course, network in the chat pod. In addition to the Adobe Connect platform, we are offering live YouTube streaming. So go ahead and invite your colleagues. However, please note that only those attendees who register and attend via Adobe Connect and log into Room 1 will be able to submit questions to our speakers. We are extremely pleased to announce that this two-day conference offers a total of 9.75 contact hours for physicians, pharmacists, and nurses. The CEs are available to all real-time attendees who self-identified as healthcare professionals during the Adobe Connect registration process and who attend via Adobe. Please note the emphasis on real-time and Adobe Connect. No CEs can be given to those who attend via YouTube or for post-event viewing. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single supporting company. CE faculty are required to disclose to the attendees when products or procedures being discussed are off-label, unlabeled, not FDA approved, and any limitations on the information that is presented. We have a spectacular lineup of speakers, and the planning committee, speakers, and CE consultation and accreditation team have nothing to disclose. Participants must attach to their attendance and complete the final activity evaluation via the CE portal. For multi-day activities such as this, participants must attest to the attendance and complete a faculty evaluation for each day. Final activity evaluations must be completed within 2 weeks after the activity, no exceptions. So in this case, the deadline for this event is December 21st, 2023. We apologize in advance, but we are unable to make exceptions to that deadline. Pharmacists should note that failure to provide your correct NABP and date of birth information in the required format may result in the loss of credit for this activity. Your NABP profile number should be the 6 or 7-digit profile number assigned by the CPE monitor, and your birth date should be in the month, day format. Do not provide your pharmacy license number. Please click the My Account tab and then navigate to Edit Contact Information to verify that your information is correct. Physicians and nurses may then view and/or print their statement of credit. Pharmacists should log into the CPE monitor 8 weeks after the last session of the activity to obtain their CE credit. Please allow NABP enough time to get this documentation into your profile. To simplify things, each night SBIA will send one email to the attendees of that individual event day. The email will have information on both the CE claim code and the survey link. I strongly encourage you to complete the evaluation, the CE evaluation, upon receiving that email before the holiday season starts in earnest, as we don't want to get that email lost, and we will not be able to assist you if you miss the deadline of 12/21/23. Okay, as we set up for this poll here, let me expand my screen so I can see the results. As we set up for this poll here, which I try to do each conference, I know that some of you may have been with SBIA for quite some time, perhaps even going back to 2010 when we first started. And I would love to say that many of you are also first-time attendees. A special welcome to the first-timers as we work towards expanding our audience base and increasing our portfolio of SBIA events. So to break the ice, get us thinking this early morning where some of us may be, I have the following question. Please participate if you can. And the question is, how many SBIA conferences have you attended? Your choices are A, this is my first one, B, 2 to 5, C, 6 to 10, D, more than 10, E, most since 2010. Okay, let's see. Numbers are still coming in. I'll give it a few more seconds. Love it. I love it, love it, love it. Oh, hmm. Okay, it looks like it's kind of settling here. So it looks like very first one, we have 68% of those people who responded. That is fantastic. Then 2 to 5 events, 20%, 6 to 10 events, 5%, more than 10, 6%. Thank you so much. And we do have 2 individuals who said they attended almost all since they started in 2010. So congratulations. For those of you who selected A, this is your first time. This is a superb event to attend as your introduction to SBIA. We encourage you to attend many more, and the next time you'll be able to select B. And thank you for those 2 individuals, now 3 individuals who have been with us almost since the beginning, and to everyone who has been with us before. Thank you. Okay, I hope we had some fun with that. As of this morning, we have over 10,100 registrants representing 130 countries. Over 52% of these registrants have self-identified as small businesses. And of those, over 32% report having 1 to 9 employees. These stats continue to be extremely reassuring to us at FDA as they confirm that we are indeed reaching the small business sector of our audience. And they also validate our mission and the very existence of the SBIA program itself. What you're seeing here is a snapshot of a geo map for today's event. And this map is always quite invigorating to us here at FDA, for it provides a very powerful visual as to the impact and extent of our outreach. And for some more fun, you can actually click on this image, which will take you to your browser where you can zoom in and have a closer look at our global participation. I invite you to try it, zoom in on your geographic area and see if anyone else is attending in your immediate city or neighborhood. Okay, so moving on, this slide here provides the QR codes for our social media avenues. You can scan them with your phones and be taken to the appropriate platform. And if you miss it now, they will also be advertised across the screen banner during the breaks. Let's talk about some details and logistics. In this, our ongoing environment, the event is 100% virtual. And over the course of these 2 days, we will have a total of 14 speakers, not to mention our organizers and technical staff, all working from our own homes or offices, as the case may be. As such, there may be some internet issues, but please bear with us. And if in fact we encounter those, we will try to get them resolved as quickly as possible. And speaking of tech issues, here is the link for a one-stop shop for answers on your technical questions, for downloading the files, accessing the survey link to obtain the certificate of attendance, and so much more. This is the same page that you used this morning for your online access to the event, so please bookmark that page as you will need it over the next 2 days. If you have any technical questions about this conference, please check there first, and you will most likely find a solution. Again, I'm inviting you to submit your questions to us so we can have a vibrant and productive Q&A segment, which is so crucial to these events. As you know, one question from one person may be applicable to so many other people, and everyone benefits from those questions. So do not hesitate to submit your questions. It's a learning opportunity. We encourage you to take full advantage of the expertise we offer here, and we will answer as many questions as time allows. So when you do submit your question, please identify the individual presenter by prefacing your question with the speaker's name as shown here. This will assist us greatly in triaging the questions. Also, during each presentation, there is a link on the right side of the pod where you will see the speaker's headshot, and it says download the slides. Note that this download link is active only during the real-time presentation. If you go and review the recording, that link will not be active for you. There is also a link to speaker evaluation, and we encourage you to take the time to complete that evaluation, as your feedback is much appreciated by the individual speakers. In addition to the speaker evaluation, we have a final event evaluation. Upon completion of this survey, used interchangeably with evaluation, An attendee can then download a certificate of attendance, which may be used in support of RAPS, SOCRA, ACRP, and SQA credits. Now, the trick is that the certificate must be downloaded immediately after completing the survey, otherwise that opportunity is lost. Now, even if you do not want or need a certificate, please take a few minutes to complete the conference evaluation, as your feedback will help guide the design of a future event. Note that both the evaluation and certificate are only available for 2 weeks. So again, the deadline date is December 21st. The survey link will be posted on a one-stop shop page this afternoon. And as a reminder, all the sessions are being recorded. It will take us about a week or two to process the recordings, but you will soon be able to access them from SBIA's homepage. I sincerely hope you have already bookmarked our homepage, which is fda.gov/CEDAR-SBIA. In the interim, the YouTube link for both days will be posted on the One-Stop Shop webpage at the end of the day. And that will also facilitate people of various geographic time zones. To simplify things again, repeating, SBIA will send our thank you email to all the Adobe attendees each day post-event, and that email will have the server link, the CE code, and should you miss the server link over the next 2 days, you can look for it in that email as well. Let's take just a few minutes to highlight our upcoming events, many, if not all, which will be of interest to this clinical trial research audience that we have here over the next 2 days. December 12th and 13th, that's next week, we will have our Navigating Complex Waters, a deep dive into FDA drug interactions, guidances, and resources. These are 2 webinars, one on each day. And they will discuss the importance of assessing the drug interactions involving therapeutic protein products, interactions caused by change of gastric pH level, and interactions affecting combined oral contraceptives. Then on January 18th, we will have a deep dive FDA's Model Integrated Evidence, MIE, industry meeting pilot program for generic drugs. Here, FDA will address considerations and expectations when meeting with FDA under the industry meeting pilot, MIE program, and we will also discuss the types of potential topics that will be granted under the pilot program. On February 13 through 15, we will host a joint FDA, MHRA, and Health Canada Good Clinical Practice and Pharmacovigilance Compliance Workshop. And now this is my personal invitation to you to join us in person as we resume our hybrid meetings. It would be so wonderful to have that energy and that excitement of meeting in person after so many years. This 3-day workshop will focus on global clinical trials and good clinical practice, bioequivalence, and pharmacovigilance in the post-pandemic world, and definitely should be of interest to this audience. Check our homepage, fda.gov/CEDAR-SBIA, seen here, for upcoming events. And we also invite you to visit our CEDAR Learn webpage, where you can link to SBIA tutorials, along with other educational resources, many of which offer healthcare professional CEs. And that website is fda.gov/CEDAR-Learn. Now, this slide here is special in that it invites your participation in a public workshop entitled Enhancing Adoption of Innovative Clinical Trial Approaches. Again, of great relevance to this audience, I would think. The workshop is hosted in partnership with the Duke Margolis Center for Health Policy and occurs on March 19th and 20th, 2024. Please scan the QR codes here and you will be taken to the website for more information on the workshop itself and also on the comment process for discussion at the public meeting. Now, this slide here gives attention to a short video which will give you an idea of our SBIA resources and the various educational materials that we offer. It will be available to play during the lunch breaks. At this point in time, I would like to acknowledge those individuals instrumental in making this event possible. Many are listed here, but so many more deserve credit as well. A special thanks to Millie Dugal, Leonard Sachs, and Kimberly Smith, the planning committee, for all the hard work in designing the agenda, recruiting the faculty, and coordinating all the details. Definitely our heartfelt appreciation to the faculty for their commitment and dedication in sharing their expertise with us. Most of all, our acknowledgement and thanks to you, the audience, who took time out of your busy schedule to attend this event and learn, regardless of the inconvenience of the various time zones. And we do not take this commitment lightly, for without you, this conference would not be possible and definitely would not be the success that it is. I am so proud again of the success of this program. I just want to say we have had over 10,000 registrants, so thank you so very, very much. A special recognition and introduction to my SBIA colleague, Captain Ray Ford, who will assist as an SBIA host over the next 2 days. Okay, now I'm sure you're ready for us to launch fully into today's agenda. And as a reminder, our speaker bios are all available on the One Stop Shop website. But for now, I'm just going to give an abbreviated introduction to the mastermind and visionary behind this workshop, Dr. Leonard Sachs. Dr. Sachs joined the FDA in 1998 as a medical reviewer in FDA's Center for Drug Evaluation and Research's Office of New Drugs. Subsequent positions include an acting director of the Office of Critical Path Programs and associate director for clinical methodology in the Office of Medical Policy. In this capacity, he has led efforts to support novel approaches to clinical trials using the use of electronic technology. Besides his involvement in the design and analysis of clinical trials, he maintains a special interest in tuberculosis and other tropical diseases and has published and presented on these topics. Please help me to welcome one of my most favorite people, Dr. Leonard Sachs. Thank you.
Thank you very much for such kind words of introduction, Brenda. So again, I'm Leonard Sachs from the Office of Medical Policy in the Center for Drug Evaluation and Research in the FDA. And I'm delighted to welcome you all to this 2023 investigator training course. At FDA, we've long recognized that clinical investigators and others involved in the trial enterprise are critical partners in development of medical products. We rely on you for all the clinical data we receive to support the approval of new medical products, and we are tremendously appreciative of the contribution you make to medical science and to the health of our society. Recognizing this, we've considered it really important to reach out to you over the years and to share our experiences as regulators in this interactive forum. As many of you know, we've been running this training course for well over a decade, and we've included lectures on non-clinical drug development, animal studies, toxicology, chemistry and manufacturing controls, clinical trials, statistical analysis, drug safety, ethics, and regulations. As we did last year, we decided to do an online course this year. These will be lectures tailored to a virtual environment. So we've limited this to approximately 2 4-hour sessions today and tomorrow. And the idea is to cover the most topical areas of interest to investigators. Today, we'll focus on selected topics related to the design of clinical trials. And tomorrow, we'll look at some of the non-clinical and the early clinical data that you may come across in investigator brochures. We will also discuss the responsibilities of investigators and investigator site inspections. By sharing our experiences inside FDA, we're hoping to provide you with an insight into FDA requirements that will give you as investigators greater confidence and expertise in conducting trials, and that it will improve the clinical trial enterprise in general. We're thrilled to see the enormous interest in the course with more than 10,000 registrants from around the world. I was fascinated to see all the comments in the chat coming in from every country around the globe. It reminds us that the development of new drugs and medical products is at the heart of medical communities everywhere on the planet. I also want to thank our speakers in advance. They're all colleagues of mine at FDA who have generously given their time to participate and prepare these presentations. I will apologize in advance for not being the most technologically gifted, but I hope we will be able to give you a satisfying experience as free of glitches as possible. And during the panel sessions, we hope you will send in your questions to make this as interactive as we can. So without further ado, I'd like to introduce our first speaker, Kim Smith. Kim is a dear colleague of mine, a nephrologist who's been at FDA for many years, and she's joined our faculty. She will be helping me with the moderation of some of the panels, and I'm going to ask her to kick off with a discussion of FDA structure and mandate. Thank you, Kim.
Thank you for that introduction. For the next 15 minutes, I'm going to provide a quick overview of FDA structure and mandate to provide some additional context for the rest of the course. So the objectives of my talk are to provide a brief history of the FDA, to describe the current mission of FDA, and to introduce FDA's legal and regulatory framework for drugs and biologics. So to start with a brief history of the FDA, I think it's important to recognize that the agency was built on a legacy of public health failures, so unsafe, ineffective, counterfeit, or adulterated drugs, as well as some unethical practices and fraud. The image on the right is an advertisement for Mrs. Winslow's soothing syrup, which was marketed in North America and the United Kingdom in the 19th and early 20th centuries as a cure-all medicine for fussy babies. But the primary ingredients were morphine and alcohol, and thousands of children are believed to have died from overdoses or from morphine addiction and withdrawal. The lower right is an image from the New York Times. This is on the Tuskegee syphilis study, which was started in 1932 by the U.S. Public Health Service working with the Tuskegee Institute. It was a study to record the natural history of syphilis. Participants were not consented and were told that they were being treated for bad blood. By 1943, penicillin was the treatment of choice for syphilis and was widely available, but the participants were never told about this or offered treatment. In 1972, the study was exposed when an article was published by the Associated Press, but unfortunately at that time, men in the study had been untreated for many years and their disease had progressed. To continue with our brief history, I'll provide an overview of just some key milestones in U.S. food and drug law. So starting with 1848, we had the passage of the Drug Importation Act, which required U.S. Customs Services to inspect entry ports for the entry of adulterated drugs from overseas. It's really one of the earliest attempts to regulate the U.S. drug supply. Moving forward to the early 1900s, at that time, diphtheria patients were routinely treated with an antitoxin that was derived from the serum of horses. The horse serum was manufactured locally, and there were no central or uniform controls. In 1901, in St. Louis, Missouri, the blood of a tetanus-infected horse was used to develop the antitoxin and was administered to 13 children who ended up dying of tetanus. This tragedy led to the 1902 passage of the Biologics Control Act, which allowed for the first controls over processes used to produce biological products, including vaccines, serum, and antitoxins. In 1906, we had passage of the Pure Food and Drug Act, which prohibited interstate commerce in misbranded and adulterated foods, drinks, and drugs. In 1912, we had passage of the Shirley Amendment. The Shirley Amendment prohibited labeling medicines with false therapeutic claims intended to defraud the purchaser, but it was a standard that was very difficult to prove. Moving forward to the late 1930s, in 1937, we had another tragedy. At the time, sulfanilamide was a drug that was used to treat streptococcal infections, and there was demand to provide it in a liquid form. So a company experimented and found that sulfanilamide dissolved in diethylene glycol, compounded the product, and sent it all over the country. Unfortunately, diethylene glycol is antifreeze and is toxic. The product killed 107 people, many of whom were children, and really provided a dramatic example of the need to establish safety before marketing a product. This tragedy led to the passage of the 1938 Food, Drug, and Cosmetic Act. The act required that new drugs be shown to be safe before marketing and really started a whole new system of drug regulation, and it was the origins of much of what we do today. In 1944, we had the Public Health Service Act. The Public Health Service Act covered a broad spectrum of health concerns and included provisions related to the regulation of biological product marketing, and really started a whole new system of drug regulation, and it was the origins of much of what we do today. In 1944, we had the Public Health Service Act practitioners. So this was our first foray into prescription products. We then have our next tragedy. Thalidomide in the 1950s and early 1960s was widely used to treat nausea in pregnant women. It became apparent in the 1960s that thalidomide was resulting in severe birth defects in thousands of children, limb deficiencies that you can see in the images on the screen. This was primarily occurring in Western Europe, and fortunately, the product had never been approved in the United States. Dr. Francis Kelsey, who was a FDA medical officer, had concerns about the amount of safety data that was available and had kept the drug off of the U.S. market. This example provided a lot of public support for stronger drug regulation and led to the 1962 passage of the Kefauver-Harris drug amendments. These amendments ensured drug efficacy and also enhanced safety controls. For the first time, drug manufacturers were required to prove to FDA that their product was effective before the product was marketed. In 1968, we had the Drug Efficacy Study Implementation, or DESI. Basically, many products had been approved between 1938 and 1962 on the basis of safety alone, and the agency had to generate a process to go back and review 4,000 plus drugs to determine whether they should stay on the market. The products that did stay on the market went through this DESI approval process. So fast forward to the FDA in 2023. The agency currently has approximately 18,000 employees, many of them scientists, and the most recent numbers are 11,000 plus of these 18,000 employees are in science fields. The agency oversees the safety of more than $2.1 trillion worth of food, tobacco, and medical products, and this accounts for 15 cents of every dollar spent by U.S. consumers. The figure on the right shows that the largest program by far is a human drug program, followed by foods, and then devices and biologics. So I'm going to shift focus a bit to provide some information about the legal framework that guides the work that we do at FDA. Our work is grounded in statute with the Federal Food, Drug, and Cosmetic Act and Public Health Service Act providing our statutory authority for oversight of clinical investigations to evaluate safety and effectiveness. The laws are further explained through regulations that the agency issues. They can be found in the Code of Federal Regulations. This is how we implement the statutory authority over the conduct of clinical investigations. A few key sections. Title 21 is Food and Drugs, and Chapter 1 addresses the Food and Drug Administration. Subchapter A provides some general provisions. This is where you'll find information, for example, on informed consent and institutional review boards. Subchapter D addresses drugs for human use, and Subchapter F addresses biologics. FDA guidance is also issued regularly by the agency, and FDA guidance is intended to be advisory. It's intended to assist regulated entities in complying with the regulations and to help them to understand FDA's current thinking on a topic. There are numerous guidances, many of which are relevant to investigators, others might be more relevant to sponsors, and if you're interested in whether there's guidance on a particular topic, the link at the bottom provides the search location. So much of the agency's work revolves around review of submissions to certain types of applications. The first is the investigational new drug application, or IND, and then you have the new drug applications, NDA, or biologics license application, or BLA, and I'll talk a little bit more about each of these. So current federal law requires that a drug be the subject of an approved marketing application before it's distributed across state lines. Because the sponsor probably will want to ship the drug to clinical investigators in many states, it needs to seek an exemption from that legal requirement. And an IND is the means through which they technically obtain that exemption from the FDA, which allows them to ship the product across state lines. This, of course, allows them to initiate clinical studies in humans. There are three main types: investigator-initiated INDs, which I think would be the most common in most situations where you're studying a drug to meet an approval standard. There are also emergency use INDs as well as treatment INDs. There are two categories. There are commercial INDs. These are the ones that are initiated by a sponsor who intends to eventually seek some sort of a labeling change or approval of a product related to the studies that are conducted under that IND. For studies that are being conducted more for academic or research purposes, there are also research INDs. All INDs must include preclinical data, manufacturing information, as well as clinical protocols and information on investigators that will allow the agency to evaluate whether the study that's proposed is reasonably safe to proceed. Not all studies of drugs need to be conducted under IND. There are exemptions for specific types of studies. So first, the product has to be lawfully marketed in the United States already. This cannot be a new product that is not already on the market. The study cannot be intended to support a new indication or a significant labeling change for the drug. The study itself cannot involve a route of administration, a dose, a patient population, or some other factor that would significantly increase the risk to participants in the study. And the investigation needs to be conducted in compliance with other regulations for instance, institutional review board, informed consent, and promotion. When the agency reviews these INDs or reviews any submission to an IND at any point in a development program, one lever we have if we have concerns about the study is to issue a clinical hold. This is an order that's issued by the FDA to delay a proposed clinical investigation or to suspend an investigation that is already ongoing. When we issue a clinical hold, no new subjects can be recruited or given the drug, and the patients in the study need to be taken off therapy unless they're permitted to continue by FDA in the interest of public patient safety. For instance, if stopping the drug abruptly would be harmful, there can be a plan to manage that in particular situations. Not all clinical holds are complete. There are partial clinical holds. For instance, if you have enough data to support lower doses but not higher doses, there may be a clinical hold on administering higher doses. Or for instance, if you have enough data to support studies in adults but not children, the studies could be allowed to proceed in adults while there's a clinical hold on studies in children. So what are the grounds for a clinical hold? For phase 1, 2, and 3 studies, the agency could find that there is really unreasonable and significant risk of illness or injury to participants in the study. There may not be enough information to understand the risk, so there may just be more information needed before a particular study or part of a study could move forward. The investigator brochure, which is intended to provide information to investigators on what is currently known about the drug, may be misleading, erroneous, or incomplete and need to be corrected to make sure that information on the drug is being accurately and completely portrayed to investigators who are enrolling patients in the study. The clinical investigators may not be qualified to conduct the investigation. And then lastly, if there is a disease that is life-threatening and affects both genders, but one gender, often women, are being excluded, that could be another ground for a clinical hold. For phase 2 and 3 studies, all of the above apply, but in this case, the agency can also put a study on clinical hold if the protocol is believed to be deficient in design to meet the objectives of the study. Moving on, once you've conducted all of your studies under IND, it comes time for a marketing application. So as I mentioned, there are two types of marketing applications, depending on whether you're a drug or a biologic. There's a new drug application, which comes under the Food, Drug, and Cosmetic Act, or a biologics license application, which comes under the Public Health Service Act. Both of these provide for submission of information that's needed to support marketing approval of a new product, or to support provide additional information and claims for an already approved product. So the key decisions before us when we get either an NDA or a BLA are whether the drug is safe and effective in its proposed uses, and whether the benefit of the drugs outweigh the risks of the drug, whether the labeling is appropriate and what it should contain, so providers who read the label understand how to use the drug and who it would be beneficial in. And then finally, whether the manufacturing methods used to maintain the drug's quality are adequate and that the methods preserve the drug's identity, strength, quality, and purity. And so at the end of the day, if we find all of these to be true, we will approve a drug and it will be legally marketed at that point. I'll move on to the challenge question. So the first challenge question is for a drug that is lawfully marketed in the United States. An investigational new drug application is never required to conduct a clinical investigation using the drug. Is this true or false? So this is false. Not only does a drug have to be lawfully marketed in the U.S., but to be exempt from IND requirements, the study can't be intended to support a new indication or significant labeling change. It can't involve a route of administration, dose, patient population, et cetera, that would significantly increase risk. And the investigation needs to be conducted in compliance with other regulations, such as IRB and informed consent requirements. So challenge question number 2, which of the following is not a reason for a clinical hold? A, the investigator brochure is incomplete. B, there's insufficient information to assess risk. Or C, the investigation of a drug for disease that manifests in childhood excludes children. Or D, clinical investigators are not qualified, which is not a reason for a clinical hold. So C is not a reason for a clinical hold. So just to briefly summarize my talk, FDA has been working to promote and protect public health for over 100 years. It's one of our oldest consumer protection agencies. The laws, regulations, and guidance provide a framework for the development and approval of drug and biologics and really frames the work that we do day to day at FDA. INDs are the mechanism through which FDA evaluates whether clinical investigations are reasonably safe to proceed. And NDAs and BLAs are the mechanism for our oversight of the U.S. sale and marketing of drugs and biologics. Thank you for your attention, and I'll be back a little bit later during the question and answer period.
Thank you for the great presentation, Captain Smith. This is Ray Ford, serving as your host for the remaining sessions. Moving into our next presentations, and the first one is on the basics of clinical trial design on design, population, intervention, and outcomes by Dr. Fred Senatore, lead physician, Division of Cardiology and Nephrology, Office of Cardiology, Hematology, Endocrinology, and Nephrology in the Office of New Drugs, CEDAR. Next, we'll learn about new trends in trial design and decentralized clinical trials, digital health technologies, and real-world evidence by both Dr. Leonard Sachs and Dr. John Concato, Associate Director of Real-World Medicine, Office of Medical Policy in CEDAR. This will lead us into our first Q&A panel. Please join me to welcome our next presenter in this session, Dr. Senatore.
Hi, my name is Fred Senatore. I'm a lead physician in the Division of Cardiology and Nephrology with the Food and Drug Administration. And today I'm going to talk about the basics of clinical trial design, specifically 4 elements: design, population, intervention, and outcome. Now, this talk is designed for clinical investigators. Many features of the topics in this presentation are usually a sponsor's responsibility. However, as a clinical investigator, you are responsible for enrolling subjects who have the disease of interest. You are responsible for not committing protocol violations. You are responsible for successfully migrating data to the sponsor or contract research organization. You're responsible for assuring subject safety, and you are responsible for assuring that each subject enrolled has been fully informed of the risks via a documented informed consent. Now, questions that come up should be asked by you, the investigator, if you feel the protocol is felt to be harmful to the subjects or felt not to be properly designed to evaluate a clinically meaningful primary efficacy endpoint. You are encouraged to question the sponsor, either individually or at the investigator meeting. Now, I'm going to talk about 4 learning objectives today, each with respect to the title of this talk. With regard to design, you will be able to appreciate what all the trial designs have in common. With regard to population, you will be able to identify the appropriate patient population for the hypothesis under investigation. For the intervention element, you will understand the various protocol designs and appreciate factors to consider in the design of clinical trials. And finally, for the outcome element, you will be able to describe primary and secondary. Now let's go to design. Pursuant to the Code of Federal Regulations 21 CFR 314.126, and I ask that you remember this particular statute, CFR 314.126, because it codifies the requirements for an adequate and well-controlled clinical trial. The trial must be adequate. That is, the trial must be designed to be able to discern a purported treatment effect based on a pre-specified criteria for success, and it must be well controlled. Usually a randomized trial is defined as a well-controlled trial that attenuates baseline imbalances of patient characteristics that could confound the results and jeopardize data interpretation. So the key message for the common trial features are that the data from the trial must be interpretable with a discernible treatment effect for drug approval. The trial design should include a pre-specified criteria for success. That is, the acceptability of a type 1 or false positive error, as well as a type 2 false negative error, the limits and boundaries where we might accept the type 1 and type 2 error. And as an investigator, you should speak up if you feel that the trial does not address a clinically meaningful issue or that the trial is not properly designed to meet the statutory requirements. Now let's go to the population. There are three types of populations. There's the intention to treat population, that is all randomized patients regardless of treatment. There's the modified intention to treat population, that is all randomized patients who took at least one dose of drug. And then there's a per protocol population, that is all randomized patients who followed the protocol exactly as written. Now, the ITT population is the preferred population that you investigate from the FDA perspective. The ITT analysis preserves the prognostic balance afforded by randomization, thereby reducing any risk of bias that may be introduced by comparing groups that differ in prognostic variables. The modified intention to treat population is tolerable. It's not preferred, but we normally accept it. The risk here is excluding subjects that could introduce bias and lead to misleading results. The per protocol population is seldom endorsed. By excluding subjects who deviated from the protocol, it introduces attrition bias, in which groups of subjects being compared no longer have similar characteristics. Now, with regard to the strategy for inclusion criteria, you would like to enroll subjects that are not too sick. You want to avoid enrolling subjects that are so sick that no matter what you do, it won't work. And then at the same time, you don't want to enroll subjects who are minimally sick. You would like to enrich the subject population with some risk factors to increase the chance of a patient experiencing an endpoint. So you want to optimize your selection strategy to not enroll subjects too sick or too well. But in the process of doing so, we need to be careful about regression to the mean. So a word about regression to the mean. One may select a subject experiencing either a bad day or a good day at baseline, that is, the tail ends of intra-subject variability. Such a subject may revert to their average level of illness during the trial. When that happens, the drug could be erroneously seen as effective if the subject had a bad day at baseline. Therefore, you're committing a type 1 error. On the other hand, a subject having a good day at baseline may empirically decrease the treatment effect of the drug. You're committing a type 2 error. Now, there's some remediation strategies to avoid regression to the mean. You should measure baseline values multiple times. Also, you should consider remote therapy monitoring, as it would apply to any eligibility criteria with intra-subject variability. Remote therapy monitoring may also capture extreme values beyond the tail ends that may not have been captured because of an exclusion criterion, thus avoiding potentially making a mistake that the treatment effect is being observed or an adverse effect is being observed beyond the intra-subject variability for the index disease presentation. The best defense here is a control group in a randomized trial. Now, a word about subgroups. The subgroup population is that which examines a particular characteristic like age, gender, race, ethnicity, region in the world, smokers, history of diabetes, or a history of myocardial infarction. The key questions that one should ask are the results the same for each subgroup? Are the results of the trial driven by only one subgroup? Is there a subgroup for which the drug is harmful? Now, there are two subgroup categories that you as the investigator should be cognizant of. The first subgroup category is one where the subjects are independent of any unique pathophysiology associated with or at increased risk for the index disease or primary efficacy endpoint. That means you're evaluating this subgroup for curiosity's sake. The second subgroup category are patients with unique pathophysiology that might matter to the treatment effect. These two groups should be treated separately when controlling for false positives. For the subgroup that might matter to the treatment effect, it is advised to have a pre-specified expected treatment effect prior to data unblinding. Now, with regard to subgroup analysis, a positive trial driven by one region, one country, one site, but negative everywhere else is still positive. A negative trial, but with a strongly positive subgroup, is still negative. Now, one must be careful about multiplicity when studying many subgroups for a possible claim. And the reason is the more tests you do, the greater the probability that you're going to be committing a type 1 error, a false positive. And this study was done 17 years ago, which demonstrated that fact. Now, with regard to the population element of this talk, the key messages here are data analysis should be conducted on the intention to treat or the modified intention to treat population. The intention to treat population should be selected to optimize the treatment effect of the intervention. One bit of caution here is that the label may be restricted to the ITT population if selection was based on a narrow severity spectrum rather than the broader population with various levels of index disease severity in the market. One must be cognizant about regression to the mean when enrolling subjects. One must also be cognizant about subgroup categories as pertains to a group that matters to the treatment effect. A positive subgroup in the setting of a negative trial does not constitute a basis for a claim, and testing too many subgroups will produce a false positive result. Subgroup analyses are therefore inconclusive. Now let's go to intervention protocol designs. Before we do that, I want to say a word about open or blind studies. An open label trial is one where everyone knows which patient is receiving what treatment. This may be a randomized control trial, or it may be uncontrolled. The pros of an open label trial is that it's easier to conduct, especially when blinding is not feasible. It's useful when comparing two approved drugs of similar characteristics, where each drug can be equally prescribed. It is useful in long-term extension trials to evaluate safety or persistence of effect. The cons of an open-label trial is intuitively obvious. It includes the possibility of bias. Patients or investigators may have a preconceived notion of the safety or efficacy of the drug that might impact their judgment. This is especially true when the endpoint is a soft one, like how do you feel, as opposed to a hard endpoint like death or myocardial infarction. Now, when it comes to blinded trials, this is a case where neither the investigator nor the patient has any knowledge about the treatment that is being given. And the key element of a blinded study is a randomized trial. Now, the pros of a blinded trial is that it attenuates potential bias in efficacy endpoint assessment, if that endpoint happens to be subjective, and attribution of adverse event to a drug. So, for example, if you're concerned about drug safety, knowing a subject is on the drug when experiencing an adverse event may result in assigning causality to the drug. So there's bias here. The cons of a blinded trial is that it's costly to perform. It's a much more complicated logistical regimen. You need randomization code generation. You need a blinded study drug kit. You need to design that. You need a distribution of the study drug kits, treatment assignment, and monitoring for study drug allocation assignment. So it's a much more complicated trial. Now, there are several blinded strategies that I think we should be aware of. There's the single-blinded trial, where the subject has no knowledge of what he or she is taking, but the investigator does. There's a double-blinded trial, where neither the investigator nor the subject know what is being administered. There's the double-blinded, double-dummy type of trial. This is a situation where it's similar to a double-blinded study, but each arm in the two-arm study, for example, has a different posology, pill versus intravenous or subcutaneous therapy. In this case, both dosage forms need to have a placebo mirror. Now, a word about randomization, which should be obvious to all of us. It attenuates influence of covariates on the primary efficacy endpoint. It's important to assess the effect of the intervention on the primary efficacy endpoint while minimizing the effect of confounders on the primary efficacy endpoint by a balanced distribution of confounders in the arms. It's as simple as that. You want to know the effect of the intervention itself on the primary endpoint without any other influence. So any other confounders are hopefully equally distributed amongst the arms in the randomization process. That is why the FDA always wants a randomization trial. And if you get a protocol from a sponsor where it's not a randomized trial, you might need to think twice about this. Now let's go into some randomized trial designs. What you see in front of you right now is a randomized parallel trial. Patients are going to be evaluated if they have stable disease. And if they do have stable disease, they'll be randomized to the active agent or placebo. That placebo may not necessarily be placebo. It could be an active comparator. This is the usual approach to evaluate superiority or non-inferiority. Screening for stable disease is a strategy to increase the probability of detecting a treatment effect. That is to say, enrolling very sick and unstable patients may cause the drug to appear ineffective. That is known as extreme enrichment. So you want stable patients in your trial, and you exclude patients who are not stable. Now, you could have another type of randomized parallel trial where there's a run-in period. The run-in period usually involves placebo or drug, and this usually evaluates compliance or toleration. And this also involves potential like drug discontinuation. That means if you're studying, for example, a hypertensive drug, and the patients they're already on a hypertensive drug, you wean them off and you put them on the test drug, and then if they tolerate it, you randomize them to continue drug versus placebo. Now, let's talk about a factorial trial. This is the case where you're going to compare drug A to drug B versus placebo versus the combination of A and B. The trial hypothesis in a factorial trial is usually that the drug combination is superior to the individual drug components. This is the case where you're trying to combine two pills into one, and this would involve a factorial trial. The crossover trial, patients are randomized to treatment A or treatment B. After a treatment period, there's a washout, and then the same patients are re-randomized. Those who took treatment A are now going to be given treatment B, and those who took treatment B are now going to be randomized to treatment A. The randomization is blinded, and none of the patients know if they got A or B initially, or B or A finally. This type of trial design is usually associated with the phase 1 pharmacokinetic trial of short-term design, approximately 2 weeks. It lowers the sample size. Crossover trials allow the response of a subject to treatment A to be contrasted with the same subject's response to treatment B. Removing patient variation in this way makes crossover trials potentially more efficient than similarly sized parallel group trials in which each subject is exposed to only one treatment. Now, here comes the placebo-controlled withdrawal trial. In this particular type of trial design, there is an open-label run-in period where everyone is given the drug, and everyone hopefully experiences a beneficial treatment effect. Then the patients are randomized to continue treatment or to placebo. So those patients on placebo will theoretically revert back to their baseline characteristics, and that difference between continued beneficial effect versus reversion to baseline characteristics would be your treatment effect. The primary efficacy endpoint is a relapse of symptoms following withdrawal of the drug. This trial design has been used for hypertension and pericarditis trials. In this particular trial design, one could measure persistence of effect and disease modification. So, key messages to the design aspect of our talk is that one must pick the right protocol design to fit the program hypothesis. The gold standard is a randomized double-blind clinical trial. It attenuates influence of confounding covariates, and it minimizes potential for bias. Now, there are certain factors to consider in designing trials. The first one is the characteristics of the disease. The second one is your objective and hypothesis. The third is primary efficacy endpoint. The fourth is time frame and duration. This is important for you as an investigator. If you feel that the duration of treatment and the protocol is not aligned with what you believe to be the duration of treatment required for a treatment effect, you need to speak up about it. And there's also a cautionary situation here where the longer the trial, the greater the possibility of dropout. Therefore, as an investigator, you must have a subject retention plan as part of the trial design. Now, also as an investigator, you need to consider trial logistics. You need to streamline the execution of the trial and the acquisition of data from your site to the CRO to the sponsor's data bank. You must have a data migration plan in hand that is usually a sponsor's responsibility, where instructions should be clear to the investigators. If not, speak up. You need to take your data that you've acquired at your site and give it to the sponsor in a manner that does not jeopardize data integrity. There are ethical considerations that need to be made here. You have to ensure that patient safety is maintained. You need a safety monitoring plan, a trial stopping criteria. You need to ensure that patients are properly informed about the study, the risks, and the potential benefits. Without a safety monitoring plan, without stopping criteria, you need to inquire about it. Also, it's important to not jeopardize patient convenience. Don't create a hardship for the patients who are in your trial. Excess travel, for example, subjects bearing cost, especially for adverse events. You need to speak up if you don't think the protocol addresses ensuring patient convenience. There are statistical considerations that you need to be aware of as an investigator. The treatment effect should be discernible above subject variability on the field function survive paradigm. the sample size should be aligned with the pre-specified criteria for success. Normally, we accept a 5% chance of committing a type 1 error, that is a false positive, and we also accept a 20% chance of committing a type 2 error, a false negative. That's where alpha of 0.05 comes in for committing a type 1 error, and beta is 0.2. That is the probability of committing a type 2 error. 1 minus beta is 0.8, and that is the power of the trial. If you're not clear on this when you read the protocol, speak up. Also, the primary efficacy endpoint should be clinically meaningful. The treatment effect should be large enough to have a net benefit for the patients. Now, the key messages here on factors to consider. When considering a protocol at your site, make sure the primary efficacy endpoint is one that you and enrolling patients deem important. If not, speak up. You need to ensure that the treatment and follow-up time frame is coincident with the treatment duration required to discern a clear treatment effect. You need to ensure the protocol has an adequate safety monitoring plan, a subject retention plan, and I'll say that again, a subject retention plan, because without subjects, and if they drop out, you don't have a trial, and you need a data migration plan. You need to ensure proper informed consent, hide nothing, and interact with your IRB. Now let's go to outcome. The outcome as defined by primary and secondary endpoints. The primary endpoint is defined as the specific clinical effect you wish to test. That is either a reduction in mortality, stroke, or heart attack. The statistical analysis plan and the sample size focuses on obtaining interpretable results and being able to draw conclusions regarding the primary endpoint. The primary endpoint can be a single variable, mortality for example, or the primary efficacy endpoint can also be a composite of several components. It's important to assure that each component of the proposed composite primary efficacy endpoint have similar degrees of clinical importance, that is to say, death, stroke, and heart attack, compared to death, stroke, and hair loss. If the second set of endpoints was the composite endpoint, and the results were positive, driven by hair loss, then the trial would be positive, but for a clinically meaningless reason. Secondary endpoints are of interest, but do not constitute the basis of a trial design. Now, sometimes a clinically meaningful endpoint, like mortality, is designated as a secondary endpoint, and the trial is powered for it. And the primary endpoint is excessively powered. That happens sometimes. Mention of the secondary endpoints in a label depends on whether they were part of a pre-specified alpha conservation strategy. This will be the subject of another talk about how to preserve alpha. Sometimes a mortality benefit, even outside of the alpha conservation strategy, may provide sufficient clinical meaningfulness to warrant a label claim. Now, I'm going to say something about the leeways of Phase 2 and the rigor of Phase 3. Phase 2 is a flexible type of study. The primary endpoint can be biomarker or a putative surrogate, and usually Phase 2 is to inform on the safety profile and the posology to Phase 3. Now, Phase 3, on the other hand, the primary endpoint must be clinically meaningful within the feel, function, survive paradigm. So if you get a protocol from a sponsor, and it's a Phase 3 protocol, and the primary endpoint is a biomarker, like, for example, NT-proBNP as a primary endpoint, there's something wrong with that, and you need to speak up. So key messages for the outcome element of this talk is that the primary efficacy endpoint leading to a claim should be clinically meaningful within the feel, function, survive paradigm. When analyzing the results of the trial, evaluating a composite endpoint of several components, consider the impact of individual components going in different directions. So if you have death, MI, and stroke, MI, myocardial infarction, and the study was positive because you prevented a lot of MI, stroke was neutral, but there was more deaths in the drug arm versus the placebo, that becomes a review issue. And when you look at unblinded data towards the end, this becomes an issue of whether or not the drug can be approved. So in conclusion and in summary, from the design element, as an investigator and clinical trialist, you are responsible to ensure the trial satisfies the statutory requirements pursuant to the Code of Federal Regulations 312.126. You are responsible to maintain knowledge on the regulatory requirements to safeguard your patients when enrolling them in a clinical trial. With regard to the population element, do not enroll very sick subjects during enrichment where the drug may not be effective. On the other hand, do not enroll a minimally sick ITT population where the drug may not have a discernible treatment effect. Be cognizant about regression to the mean when enrolling subjects, and use your clinical knowledge as an investigator if you think the protocol eligibility criteria are enrolling subjects at the tail ends of variability at baseline, thus potentially impacting the results. From the intervention point of this talk, focus on patient safety by ensuring the existence of and adherence to an adequate safety monitoring plan. Understand the data migration plan and the subject retention plan and adhere to it to minimize missing or corrupt data. Keep control of your site. Maintain vigilance over the research team and correct any issues in an expeditious way. With regard to the outcome, if you, as an investigator, happen to serve on a steering committee, you should ensure the primary efficacy endpoint is clinically meaningful. That means based on field function survival and discernible by the subject above the normal variability of disease presentation. As an investigator, either on a steering committee or simply being an investigator, if you feel these things are not the case, speak up. So in conclusion, ask the sponsor if you have any questions. That's the most important thing. You, the investigator, have a significant say in the trial design. The investigator meetings are designed to ensure the protocol is consistent with the clinical practice paradigm. You, the investigator, are responsible for your patients enrolled in the trial. That is safety, informed consent, trial with a clinically meaningful outcome, and data management at your site. You, the investigator, are ultimately changing the practice paradigm for the better by bringing into the market a drug that you will have contributed to in demonstrating a beneficial treatment effect. And I want to thank you for your service to our society. This is an important role that you are taking as a clinical investigator. Now, I do have a couple of challenge questions. The first one, if a sponsor wishes to include the primary and series of secondary efficacy endpoints in the label, it is incumbent on the sponsor to develop a hierarchical analysis plan for testing each endpoint by pre-specified order in order to avoid the committing a type 1 error. I'll repeat this. If a sponsor wishes to include the primary and a series of secondary efficacy endpoints in the label, it is incumbent on the sponsor to develop a hierarchical analysis plan for testing each endpoint by pre-specified order to avoid committing a type 1 error. Is that true or false? I'll give you 10 seconds. The answer is true. And here's my second question. Which of the following are true? A global trial is considered positive if the entire population, that is the intention to treat population, met the pre-specified criteria for success, even if subjects in the United States did not show a benefit. B, attempts to determine specific subgroups for which the drug will benefit the most in order to better guide healthcare practitioners may increase the probability of committing a type 1 error. C, a positive trial that is driven by only one region of the world, that is to say Russia and Ukraine, which is normally the case, but negative everywhere else, disqualifies that trial from having met the evidentiary standard. D, A, B, and C are correct. E, A and B are correct. Or F, B and C are correct. I'll give you 15 seconds. the answer is A and B are correct. The reason why C is not correct, a positive trial that is driven by only one region of the world is still a positive study. So that concludes my challenge questions, and I want to thank you very much for listening to this talk, and we'll be available for questions should you have any. And good luck with the rest of the talk, and have a wonderful day.
We're going to spend the next half hour discussing some innovative trial approaches, including digital health technologies and decentralized clinical trials. I think we all recognize that there's been significant progress in biosensor development and in communication technologies, and the opportunities for remote collection of data are quite remarkable. These technologies, as we know, are playing an ever-increasing role in clinical care, and digital health technologies and electronic communication platforms can obviously perform a significant role in the evaluation of medical products. Why bother with these technologies and approaches? This is a child with Duchenne's muscular dystrophy. As many of you know, Duchenne's is an inherited condition in young boys. It results in progressive muscle weakness, eventually tragically resulting in death, usually from respiratory failure when patients reach their teens. And this boy is participating in a clinical trial. In order to get to his trial visits, he has to be taken out of school. His mother has to take time off work. A flight has to be booked. They have to get onto the flight, transport the wheelchair, get into a hotel, and eventually get to the trial site. A very arduous and disruptive process. When they get to the trial site, this is what gets done. This is the 6-minute walk test, and the child is supervised, and the child is told to walk as far as he can for 6 minutes, and the distance is measured. It certainly leaves us scratching our heads. This is a picture of an accelerometer. These are tiny devices. Typically 3 of them are used together in mutually perpendicular directions, and they provide a very good recording of all sorts of movements, including, of course, steps. Accelerometers are versatile tools for Duchenne's and for many other diseases. Accelerometers are used all over. We have them in our cell phones, our smartwatches, Fitbits, and so on. They can be very helpful in measuring steps or other activities taken by a Duchenne's patient with no need to depend on snapshot measurements or clumsy tests like the 6-minute walk distance. They can measure novel aspects of functions such as stride velocity, and in Duchenne's, the 95th percentile of stride velocity has been considered a promising measure to assess drug efficacy, and finally, comparisons with baseline measurements can be made. They capture objective data on functionality, which has traditionally been challenging in clinical trials. They may be useful in neuromuscular and cardiorespiratory diseases, muscular dystrophy, Parkinson's disease, heart failure, chronic obstructive pulmonary disease, and pulmonary hypertension. And they have a potential role in neuropsychiatric diseases, depression, ADHD, and schizophrenia, which are difficult to measure using current methods. And here are a couple of publications on the use of accelerometry. The first is by the European Medicines Agency, talking about quantification of stride velocity as an endpoint in studies in ambulatory Duchenne's muscular dystrophy. And the other one's a report in the New York Times, where they report wearables track Parkinson's disease better than human observation. As far as biosensors go, they measure clinical features, and these may either be discrete events like a step, a breath, a cough, a pulse beat, a seizure, a tremor, or forced expiratory volume, or they may be continuous readings like we get from a continuous glucose monitor, glucose readings, pulse oximetry, temperature readings, electrocardiograms, and blood pressure. Biosensors begin as a transducer, an electronic device that transforms a physical finding like acceleration or temperature or pressure into an electrical output. As you can see, there are a number of different types of transducers that we see in use: galvanometers, accelerometers, photoelectric cells, electrochemical sensors, and thermocouples all detect different physical properties. The output, as I mentioned, is some electric output, which might be either voltage, current, or impedance. And if these are intended to measure a clinical feature, for example, things like heart rhythm, walking, scratching, tremor, or blood oxygen saturation, etc., an algorithm is needed to process the data. Sometimes the algorithm includes calibration curves, which are needed in electrochemical sensors. And the result of the sensor and the algorithm is a clinical digital health technology. As you can see there, a cardiac monitor, an activity tracker, pulse oximeter, continuous glucose monitor, and a digital thermometer. Can we rely on the DHT? Verification is generally a process that's conducted in a lab, and its intent is to determine how accurate the DHT is in measuring the targeted physical feature, for example, acceleration or temperature. Is it reproducible in different environments, different temperatures, different humidity, and so on? Validation is usually done in the field, and the intent of validation is to show how suitable the DHT is for its intended use. Are the data recorded by the digital health technology in the patients the same as the data we would report if we were looking at the patient? For example, steps in a patient with Duchenne's muscular dystrophy, or steps in a patient shuffling with Parkinson's disease. Is the result affected by how the patient wears or uses the DHT? And are there other things that a patient might do that would be misinterpreted by the DHT? For example, tapping a foot or riding a bike. The suitability of DHTs for use in a clinical trial is obviously very important. Are they ugly or elegant? Are they easy to put on? Are they easy to operate? Are they comfortable to wear for the required duration of the study? Is the battery life convenient? Can data be synced? And can patients bring their own devices? I wanted to end this discussion on digital health technologies by mentioning that mobile applications are also considered DHTs. And here you can see a couple of examples: a patient-reported outcome on a cell phone camera that's used to capture a lesion, a coordination test in Parkinson's disease on the top right. This test has 2 buttons on the cell phone, and the patient's required to alternatively tap each of the buttons to test their coordination. And finally, things like visual tests on a cell phone. What about endpoints based on DHT measurements? Justification of the endpoint is important as a clinically meaningful measure of drug effect. When we think of the endpoint, it has various components. The first is what is being measured, for example, steps. What is the time window of observation? Let's assume this is 4 weeks. And what is the formula for the response in each patient, which in this case may be changed from week 1 to week 4 in the average daily step count. Another question is whether the endpoint is a clinically meaningful measurement of drug effect. And this can be established by comparison with existing benchmarks of performance, for example, things like the Unified Parkinson's Disease Rating Scale or other patient-reported outcomes, the 6-minute walk distance. It would also be very important to get input from patients, from caregivers, professional societies, disease experts, and regulators regarding the meaningfulness of the endpoint. I put the slide in just to remind ourselves of the differences between validation and verification versus justification of an endpoint. I think it's important to remember that validation and verification are technological assessments. They address how well the technology measures the clinical feature of interest. On the other hand, justification of an endpoint or a clinical outcome assessment is a clinical issue. It addresses how well the clinical feature of interest represents a meaningful response to treatment, and it has nothing to do with the DHT or with a method of measurement. Where are digital health technologies likely to be of use in drug development? And what I've done here is summarize the endpoints that were used in registrational trials between 2015 and 2020. You can see that there were 218 new drug applications during this period, and about 30% of them, shown in beige, relied on a biomarker, for example, a chemistry measurement like hemoglobin A1C, pregnancy test in contraceptives or GFR, hematology tests, pathology tests, and microbiology tests. About 22% relied on imaging. A lot of these were cancer trials where imaging was part of progression-free survival assessments, but there were other applications that looked at bone mineral density or vertebral fractures and spleen volume. About 7% focused on physiological or functional measurements. These included things like the 6-minute walk distance, normal sinus rhythm, forced expiratory volume, or sleep studies. 22% relied on clinical events or clinical signs, and these were generally prophylactic studies. There were studies designed to prevent things like death, hospitalization, major adverse cardiac events, or multiple sclerosis relapse. And finally, 32% relied on clinician-reported outcomes or patient-reported outcomes. And these were things like the Hamilton Depression Rating Scale, rheumatology scales, psoriasis severity indices, seizures, sleep, prostate symptoms, and other scores. So there are many types of endpoints where mobile technology tools may play a role. First of all, as we saw, clinical lab measurements, continuous glucose monitoring, and pulse oximetry are some of the obvious ones. Physiological measurements like heart rate and rhythm, breathing and lung function, seizures, syncope, temperature, and weight. And finally, performance assays, things like stamina, strength, coordination, abnormal movements, sleep, and cognition. Advantages of these type of continuous readings by biosensors are richer data instead of snapshot data. For example, we can get things like the average number of steps a patient takes a day versus the 6-minute walk distance, continuous glucose monitoring versus just a single hemoglobin A1C in diabetes. They give us the ability to detect rare events like arrhythmias, seizures, or apneic spells. We can get data from patients who cannot report, for example, scratching in infants with atopic dermatitis, or sleep patterns in patients with dementia. We can get dose response information, for example, the on-off effects of treatment in Parkinson's disease, and we can also make new types of measurements that weren't possible before. For example, accelerometry measurements of gait stability that may predict falls in patients with movement disorders, measurements of coughing, sneezing, and tremor, and behavior patterns in patients with dementia or depression. DHTs can be used for things other than endpoints. They can be used for enrollment, screening, and enrichment, helping qualify a baseline disease severity and functional status. They can be used to monitor certain aspects of safety, for example, identification of rare adverse events, and they can also provide investigators with real-time access to patient safety data. They allow visualization of dose responses over the dosing interval. And finally, as far as endpoints go, their use would be most compelling in superiority studies, which show that one arm is better than another. Non-inferiority studies may be challenging to interpret. If the DHT is not working properly, it may give us the false impression that both arms are equally effective. What is our regulatory position on digital health technologies? We don't have regulations that directly address the use of DHTs in clinical trials. DHTs in clinical trials generally do not need to be approved or cleared by FDA for marketing unless they are medical devices. Our regulatory standard are the qualities of the data from a DHT adequate to provide substantial evidence of effectiveness? And finally, are the data attributable to the patient? Are there processes in place to ensure the data integrity, data security in transit and during storage? And you can refer to our guidance on the use of electronic health records and electronic signatures in clinical investigations listed below for more details on Part 11 as it applies to data integrity. Now let's turn our attention to decentralized clinical trials. Decentralized clinical trials are really a package of strategies. These are trials where some or all of the trial-related activities take place remote from the investigator at a place or location that's convenient for patients and that do not require visits to research site. The strategies include video and telemedicine visits, the use of digital health technologies, direct distribution of the product to patients' homes, the use of electronic informed consent so that patients can sign their consent at home, the use of home visits by either clinical staff or local healthcare providers, and of course the use of local healthcare providers themselves and facilities to provide some of the data used in the clinical trial. Why are regulators interested in decentralized clinical trials? Well, firstly, accessibility. This would be very important in patients with rare diseases who widely spread across the country or across the world. Second of all, for patients with mobility or cognitive challenges who have difficulty getting to trial sites. And thirdly, decentralized trials may improve the diversity of participants. For example, those who have socioeconomic challenges and cannot afford to take time off work where they're paid by the hour, cannot afford to employ child support while they're away. Obvious advantages for patient convenience, not having to get to trial sites. And then there are other efficiencies. For example, decentralized trials may avoid the costs associated with travel. They may reduce the need for physical facilities where trial-related activities are conducted. And they may also take advantage of qualified community providers who are doing a good job in clinical practice and may provide useful trial-related services as well. And then finally, our experience with COVID-19 has shown that these decentralized approaches are very important to prevent the spread of contagious diseases. Some concerns about remote data acquisition. It's important to ensure that there's oversight of trials as trial-related activities get delegated to local healthcare providers and others more remote from the investigator. Oversight has to be carefully planned. There would be potential concerns about device failure. Patient safety is obviously a primary concern. If there's any risk to the patient's health by not being close to the investigator site and not having access to other care, then that would not be a suitable environment for a decentralized trial. Patient retention has been a potential problem. Some patients just prefer the regular face-to-face visits that they have with their providers, and they may be less interested in engaging through remote technologies. Data privacy is also something that has to be ensured in these decentralized environments. Decentralized clinical trial procedures are not new. Patient diaries and interactive voice response systems have been used for years. Telephone follow-up has been common, particularly long-term follow-up in trials. In 2016, we published a guidance on electronic informed consent, which allowed consent to be done from patients' homes. In 2014, the first entirely web-based trial was conducted under IND. This was a trial looking at tolterodine for patients with overactive bladder. About 5,157 patients were registered for the trial. There were a lot of arduous procedures, including micturition diaries and numerous urine samples that were needed. And although 456 signed consent, 188 in the placebo run-in, only 18 finally were randomized to treatment. Electronic informed consent was used, and patients were randomized to receive tolterodine or placebo over a 12-week period. The outcome was determined using electronic diaries, and there were 2.4 less micturition events per 24 hours in the treated arm, as opposed to 0.8 micturition events less in the placebo arm. Treatment difference was 1.6, which was not significant statistically. And I think the lesson from that trial was that it was very important to focus on simplicity in decentralized trials and make sure that patients can comply with the requirements. Recognizing the potential opportunities that decentralized clinical trials offered to address challenges in trial accessibility, we published a draft guidance in May this year on decentralized clinical trials for drugs, biological products, and devices, and I'm going to spend the next few slides going through some of the important points raised in this guidance. So firstly, remote trial visits. Investigators can conduct trial visits remotely using video conferencing or other suitable technology, and this may make trial visits much more convenient for patients living far away, but clearly there are challenges. In many states, local regulations on telemedicine may make this difficult to implement. Physical examinations are not really possible when you're doing a remote trial visit, although the investigator may be able to supervise somebody doing the examination in the patient's room. Video photography may not fully capture the features of a lesion where you want to detect things like induration, tenderness. Patient engagement in the absence of in-person contact is sometimes a challenge. There are certain patients who prefer to see their providers in person rather than getting onto a telemedicine visit. Complex drug administration procedures are a challenge during a remote visit and may need specialized trial personnel on site. And finally, close medical supervision is really not suitable, for example, when one anticipates infusion reactions. And in these situations, remote visits are probably not appropriate. Direct distribution of investigational products. Well, the investigator should control the release of products to trial participants. Remember that the investigator is responsible for administering the trial drug. Local state laws differ on direct distribution to patients. Some of them may require locally licensed healthcare professionals or pharmacists to prescribe the investigational product. Packaging and handling are important considerations, particularly when we're dealing with unstable or labile medical products. And finally, plans have to be made for disposal of unused product. What about the use of local healthcare providers and local facilities? There are many examples where local healthcare providers or local practitioners perform tasks that are not directly related to the care of the patient. For example, an insurance company may get a local healthcare provider to do an insurance medical, or a local healthcare provider may do a visual acuity test for a driver's license. And in the same way, we believe that local healthcare providers are an important resource in clinical trials. These are qualified people in the clinical care environment, and they obviously bring their expertise to the clinical trial arena. Routine clinical activities can be delegated to local clinics or local healthcare providers. For example, things like x-rays, clinical examinations, and lab tests. Obviously, it's important to ensure that these individuals are appropriately qualified in the community. We mentioned in the guidance that tasks logs allow investigators to keep a record of who's doing what. We've had a lot of comments in the docket indicating that they didn't agree with this, and we will take a more careful look at whether this is necessary. Another important aspect is that the data has to be regularly reviewed by investigators, seeing they're not doing these examinations or these procedures in person. Electronic informed consent is already in widespread use. It allows patients to review and sign the consent at their homes. It may allow the provision of videos and graphics to make the process more informative and more easily understood. And of course, important to remember that signed consent must be obtained before enrollment of patients. Home visits are another novel approach. These can be done either by dedicated trial staff or by local healthcare providers. Mobile trial units are also being developed. These would be vans that travel in the community where trial-related procedures can take place. And these strategies are another way of extending the physical reach of the trial. Digital health technologies, as I mentioned before, these provide access to continuous or frequent data. They give us the ability to detect sporadic events, for example, seizures, arrhythmias, and falls. They allow patients to report the outcomes in real time, something that's often called ecological momentary assessments. They provide real-world environment evidence of drug effect, for example, while patients are working, exercising, and sleeping. And they also are able to provide an objective record of functionality. And for more information on digital health technologies, I refer to our guidance on digital health technologies published in January 2022. What about oversight? The investigator, as in any trial, is responsible for oversight of the clinical trial. The investigator and sub-investigators should be listed on Form 1572. As far as oversight of local healthcare providers, such as doctors, nurses, radiologists, delegated to do trial-related activities, investigators should review their reports regularly and make sure that there's no missing data or that data are not erroneous. And when patients present with unrelated complaints to local emergency rooms, neighborhood clinics and pharmacies, obviously these institutions are not part of the trial and not subject to investigator oversight. As far as safety goes, high-risk products and severe diseases may not be suitable for decentralized clinical trials. It would be important to ensure that patients can contact study staff if they run into a clinical problem. And it's also important to ensure that local medical facilities should be available for urgent care if the patient runs into a problem with adverse events or other trial-related issues. What about inspection? A site inspection would generally take place where the data are and where the study staff can be interviewed. As far as local healthcare providers, local inspections may be necessary if problems are found, and ancillary medical services like emergency rooms and neighborhood clinics would generally not be subjected to inspections, just as they are not for traditional clinical trials. A couple of other considerations. Not everything can be accomplished remotely. Many decentralized trials will end up as hybrids, combinations of remote visits and visits to investigator sites where procedures such as detailed physical exams and other in-person activities are required by the protocol. Supervision, as I mentioned, is important. Local healthcare providers and local facilities need to be supervised by investigators, and records need to be available from local healthcare facilities, allowing inspectors to recreate the trial. I wanted to finish off with a couple of thoughts about trials in clinical practice settings or point-of-care trials. These provide the opportunity to use existing clinical infrastructure, particularly when it's supported by interoperable data systems like electronic health records, and this has become an area of increasing interest. The recovery trial in the UK for COVID-19 was the poster trial for trials in a healthcare setting. In that trial, they reportedly recruited 40,000 COVID patients through the National Health System in the UK within the space of 6 weeks. Quite an extraordinary achievement. And they were able to show the mortality benefit of steroids in treating patients hospitalized with COVID with hypoxia. And this has now become standard practice. Practice settings allow engagement of large numbers of patients in short periods of time, and they reflect the effectiveness of treatment in real-world environments, and they provide accessibility of clinical trials to patients who wouldn't normally participate. So it leads me to my conclusions, which are that there are important opportunities to improve trial efficiencies, convenience for patients, and the ability to access diverse participants and rare diseases. Electronic technology has broadened the scope of decentralized activities considerably. Contacting FDA. If you're considering an innovative trial designed for a drug-specific application, you should contact the relevant review division for a pre-IND meeting. For engagement on digital health technologies or decentralized clinical trial designs that are not related to a specific drug development program, you can contact us at the email address given here, and we will determine the best forum to address this. A couple of questions. Justification of an endpoint involves ensuring that the DHT is accurate and reliable, true or false? Well, that's false. Remember that verification and validation ensure that the DHT is accurate and reliable. Justification of an endpoint is a clinical determination and has nothing to do with the method of measurement. All local healthcare providers participating in a clinical trial would need to be listed on Form 1572, the statement of the investigator. Is that true or false? And that's false. It's just investigators and sub-investigators who need to be listed on Form 1572. And finally, which of the following is not a strategy to decentralize trial-related activities? Video and telemedicine visits, direct distribution of product, electronic informed consent, visits by participants to trial sites, and the use of local healthcare providers and facilities. And clearly, from what we've been through in the lecture, visits by participants to trial sites are what we're trying to avoid with decentralized clinical trials. So that would be a strategy that is not designed to decentralize trial-related activities. Thank you.
Hello, I'm John Concato from the Office of Medical Policy in the Center for Drug Evaluation and Research, and I'll be discussing real-world evidence. As objectives and an outline of the presentation, I'll first describe the scope of FDA's real-world evidence program. I'll move on to recognize the intersection of scientific and legal and regulatory issues related to study design in the real-world evidence era. Next, we'll interpret terms commonly used for study design and drug development. And then we'll move to identifying examples of real-world evidence in drug approvals. Let's start with the 21st Century Cures Act of 2016, which prompted FDA to establish a program to evaluate the potential use of real-world evidence to support a new indication for a drug already approved or to satisfy post-approval study requirements. A draft real-world evidence framework was issued in December of 2018, and draft guidance for industry was issued in late 2021. It's important to note that our standard for substantial evidence remains unchanged, and we can also mention that commitments were met under the Prescription Drug User Fee Act 6, and there is a new Advancing Real-World Evidence Initiatives program in PDUFA 7 that started in October of this year. The 2018 Real-World Evidence Framework applies to the Center for Drug Evaluation and Research, as well as the Center for Biologics Evaluation and Research and the Oncology Center of Excellence, but not to other centers. The program involves internal agency processes, external stakeholder engagement, demonstration projects, and guidance development, although I only have time to discuss the last two of these four components. FDA has formal real-world definitions from our 2018 framework. On the left, we see real-world data or data related to patient health status or the delivery of healthcare routinely collected from a variety of sources. We often think of electronic health records, medical claims, registries, and the other sources shown. On the right, we see that real-world evidence is defined simply as the evidence regarding the usage and potential benefits, risks of a medical product derived from the analysis of real-world data. And note that randomized trials can generate real-world evidence when they incorporate real-world data. Let's now turn our attention to study design, starting with traditional terms used to describe patient-oriented research for several decades. On the left, we see descriptive studies, including case reports and case series. And on the right, we have cause and effect or analytic studies with a control or comparator group, which are usually described either as observational studies or what could be called experimental studies, but with an intact patient as unit of observation. For all intents and purposes, randomized trials are the most common experimental study design. When considering randomized studies to evaluate drug outcome associations, we see that we have patients at baseline who receive a drug or a comparator who are then evaluated for an outcome. When we ask the question whether the validity of the comparison is affected by sources of methodologic bias, we recognize that randomization promotes balance at baseline to minimize bias, and for decades, randomized trials have been the preferred method for evaluating drug safety and efficacy. This second of two design slides shows non-randomized studies where patients at baseline again receive a drug or a comparator, but this time in usual clinical practice, and an evaluation of outcome is subsequently done. Is the validity of this comparison affected by sources of methodologic bias? Well, observational studies need to address baseline imbalances to minimize bias, such as accounting for the drug of interest being given preferentially to patients who are more likely to have either better or worse outcomes. A topic related to the prior two slides is shown here. Most, if not all of you, have seen this or similar hierarchies of study design which evolved in the 1990s, designating randomized controlled trials as a gold standard study design and suggesting other study designs are not trustworthy. More recently, however, emphasis has been placed on strengths and limitations of different study design types. One view is represented by an article titled "The Magic of Randomization versus the Myth of Real-World Evidence." The article has the quote, "Because of potential biases in observational studies, such studies cannot generally be trusted. The replacement of randomized trials with non-randomized observational analyses is a false solution to the serious problem of ensuring that patients receive treatments that are both safe and effective," close quote. I'll just mention here that few, if any, people are suggesting the replacement of randomized trials. Nonetheless, in contrast, we have a misunderstanding randomized trial article that argues, quote, "Any special status for randomized trials is unwarranted. Which method is likely to yield a good causal inference depends on what we are trying to discover, as well as on what is already known," close quote. Several CEDAR staff published on this topic in an article titled "Randomized, Observational, Interventional, and Real World, What's in a Name?" The take-home points include that FDA is evaluating whether and how observational studies intended to evaluate efficacy can contribute persuasive results from scientific and regulatory perspectives. And in this context, a randomized trial versus observational study dichotomy is overly simplistic as shorthand for strength of study design to support causal inferences. Additional comments are also shown in this excerpt. To better understand the term real-world evidence, we should begin with the term big data, which first appeared in the computer science literature during the 1990s, often referring to data too large to be stored in then-conventional storage systems. It's unclear when big data became the buzzword of the day, or really what it exactly means, but one perspective is that the integration and analysis of large-scale data has always been integral to epidemiology, but what's changed is that modern technology has increased the quantity and forms of available data, as well as the speed to manipulate and merge such data. If we focus on the term real-world evidence, we should recognize that real-world is actually a non-specific modifier, and real-world data and real-world evidence appeared in the medical literature as early as the 1970s or before that even, but in various contexts. In contemporary usage, however, RWD and RWE have formal regulatory definitions as I described. So one perspective is that the older epidemiologic terms were sufficient, but the emergence of big data and the enactment of 21st Century Cures Act has led to what turns out to be sometimes confusing use of different taxonomies for study design. As just one example, when you hear real-world evidence study, don't automatically think of an observational study. You really need to hear specific details to classify the study design. Based on that historical overview, we can now talk about contemporary terms for study design. An interventional study or clinical trial is where patients are assigned to treatment by a study protocol. A non-interventional or observational study is where patients receive treatment during routine clinical care. Importantly, a study can have components of both. For example, an externally controlled trial with an interventional treatment arm and a non-interventional control arm. Those same terms are used in an article from earlier this year titled "Real-World Evidence, Where Are We Now?" You see in the top row randomized interventional studies, non-randomized interventional studies, and non-randomized non-interventional studies. We tend, however, to jump down to the next row to talk about traditional randomized trials, trials in practice settings, externally controlled trials, and observational studies, with the main body of the figure showing different design types. It's also important to note that the generation of real-world evidence does not occur when a traditional randomized trial uses real-world data only for planning purposes on the left, such as to assess enrollment criteria or to support selection of trial sites, and that it's an increasing reliance on real-world data going left to right, and we do finally catch up with the observational study category on the far right. To clarify when real-world data generates real-world evidence, this more recent article by FDA co-authors provides even more granular details. This table is from the same article, and I won't go into all the details, but you could see that the content describes when interventional studies, aka randomized controlled trials, as well as externally controlled trials, and non-interventional studies do or do not generate real-world evidence. I'm repeating this slide that was shown earlier in the talk to pivot back to FDA's real-world evidence program. As mentioned, demonstration projects and guidance publications represent two major initiatives to promote the appropriate use of real-world data and real-world evidence. The next several slides will go into some detail on these two topics. First, demonstration, that is, research projects, represent FDA's contribution to advancing the science of real-world evidence. In informal categories of improving the use of or quality of real-world data, in the middle we have studies looking at study design proper, and on the right, so-called tools to improve real-world evidence. I'll only go across the top row for the sake of time to point out that a OneSource project with the University of California, San Francisco is designed to improve the quality of EHR data at the bedside. The next slide will show a little bit more about the RCT duplicate trial emulations, wherein real-world data combined with observational study design were used to try to emulate previously completed or ongoing randomized trials. And on the right in the top row, evaluation of confounded treatment effects was a study with the University of North Carolina that looked at the available methods to assess unmeasured confounding. This slide shows a snapshot of the primary publication from the RCT duplicate project coordinated by investigators at Brigham and Women's Hospital. I'll just emphasize the first sentence that states, quote, real-world evidence studies can reach similar conclusions as RCTs when design and measurements can be closely emulated, but this may be difficult to achieve, close quote. I refer you to the article for further details. Time also doesn't allow me to do more than list the series of FDA guidance documents on real-world data and real-world evidence. The bottom line is that we're sharing our current thinking on the topics shown, which can be grouped in categories of data considerations, submission of data, applicability of regulations, design considerations, and procedural issues. We have 2 more guidances to publish, and after receiving public comments, each of the guidances will be converted from draft to final. Let's now segue to FDA's approach to evaluating real-world evidence. Key considerations are summarized here. First, whether the real-world data are fit for use. Second, whether the study design can generate adequate scientific evidence to answer the regulatory question. And third, whether the study conduct itself meets FDA regulatory requirements. A final and more recent example is a new indication for a drug called Prograf based on real-world evidence. The drug tacrolimus had been approved for prophylaxis of organ rejection in patients receiving liver, later kidney and heart transplants based on traditional randomized trial evidence, and the drug was being used widely in clinical care. For various reasons, randomized trials were not done for lung transplant, but the sponsor submitted a supplemental new drug application to FDA with a non-interventional quote-unquote real-world evidence study. The data and design were evaluated according to FDA standards, and approval for preventing rejection and death in lung transplant was granted in July of 2021. Applying the considerations shown on an earlier slide to the program approval, we see that the data came from the U.S. Scientific Registry of Transplant Recipients involving all lung transplants in the U.S. during the period shown. The design was a non-interventional observational treatment arm compared to historical controls. And FDA determined this study to be adequate and well-controlled, or highest evidence bar. Of note, the outcomes of organ rejection and death are virtually certain to occur without therapy, and this dramatic effective treatment helped to preclude bias as an explanation of results. I don't wish to leave the impression that working with real-world data to generate real-world evidence is easy. In fact, this slide highlights what can and often does go wrong in the same three categories of data sources, study design and analysis, as well as study conduct. To highlight just a few, data reliability and clinical relevance are often lacking in the first category. In the second category, confounding has been mentioned before, as well as complicated problems with the index date or zero time that I don't have the opportunity to discuss in detail. And then last but not least, under the conduct of randomized studies, the protocol and analysis should be pre-specified, and FDA requires access to patient-level data and the ability to inspect real-world data sources. In summary, FDA's real-world evidence program is advancing as outlined in the agency's 2018 framework for real-world evidence. It's important to recognize that new terminology linked to the emergence of big data and the passage of 21st century cures is often used inconsistently, and randomized trials versus observational studies is an oversimplified dichotomy. It's also true that older terms for study design in drug development are now joined by newer terms describing those same study designs. But regardless, FDA approves drugs and biological products using real-world evidence based on applicable and existing regulations, maintaining our evidentiary standards. Finally, a knowledge check. True or false, randomized trials are not within the scope of real-world data, real-world evidence at FDA. This statement is false. Randomized trials generate real-world evidence when they incorporate meaningful real-world data. The second statement, real-world evidence studies for effectiveness are held to a different, that is, lower evidentiary standard than randomized trials. This statement is also false. Regardless of the type of study design, the FDA has specific standards for judging whether a product is safe and effective for use. Thank you very much for your attention, and I look forward to the question and answer discussion period.
Fred, Leonard, and John, thank you all so much for excellent presentations. The good news is we have a very engaged audience. We've gotten several questions in the chat, so we'll see how many we can get through over the next 15 minutes or so. Hopefully you're all able to join me with the audio for this chat. It looks like we've gotten questions for several of you. Why don't we start with Fred? So Fred, one question we have, how can an investigator ensure enrollment of the right subject, with right in quotes? That is a subject who's not too sick so as to render treatment ineffective, or not too well so as to decrease the probability of reaching an endpoint.
Can you hear me? This is Fred.
Yes, Fred, go ahead.
Okay, and should I turn my camera on or leave it off?
Off is fine, thanks, Fred.
Okay. That's not an easy task to enroll the right patient. What you need to do is, during the investigator meeting or when you are reviewing a protocol, look at the inclusion and exclusion criteria. If you start seeing class, like for example in cardiology, New York Heart Association class 1 or 2, these are patients who could pretty much act normally, and that would give you a clue that this may not be the right patient to see a treatment effect unless there's a huge sample size or there's a trajectory that leads to expeditious degradation of the health status. And on the other hand, if you see a lot of class 4 subjects that are includable or included in the clinical trial, that's a clue that they're too sick. So look very carefully at the inclusion exclusion criteria. And when you enroll a patient, try to be patient, meaning try not to enroll a patient because you need to have 2 or 3 patients per month to meet your quota, and accidentally enroll patients that might fall outside of the not too sick and not too well population. So this is a careful scrutiny of clinical presentation, your evaluation of the patient status when you're enrolling them, making sure that they're just the right patient. That's easier said than done.
Thanks, Fred. And you have another question here. How can an investigator know whether or not the results of a trial may be regression to the mean? How does an investigator guard against this when the contribution to the overall sample size is small at a specific site?
Yes, that's a very good question. And how one would guess, one would never know, because you're enrolling a patient and you're in isolation. You're at one site, one investigator, one sub-investigator, one patient comes in, and you have to have a good clinical sense as to what the average presentation is in this patient population, what they can and cannot do. And when you gauge the mean status of the patient, and then you look at the patient in front of you that you're about to enroll, are they looking like it's too well? Are they too well, or are they too sick relative to what you feel is the mean? This gives you a sense that at baseline, if you're enrolling a very sick patient at baseline, they might get better because of concomitant medications or because of the natural history of variability of the disease. So again, look at whether or not your patient that's in front of you represents the tail end of a mean presentation, and that might give you a clue as to the potential risk for regression to the mean.
Great, thank you. And Leonard, we have questions for you on both DHTs and DCTs. Why don't we start with a couple on DHTs? So the first, does digital health technology have to be approved for medical use to be used as a digital health technology in a clinical trial? The example they give is an Apple EKG, or is validation of the technology for the investigator's purpose allowed for the clinical trial? For instance, comparison of a self-written app to a gold standard.
Thanks, Kim. So it's not a requirement that every DHT has to be cleared or approved. If a DHT that's used in a clinical trial is being used as a device, as a medical device, then it does have to be cleared, but otherwise not. So, for example, things like activity trackers and so on do not have to be cleared by CDRH, but they clearly have to be validated to make sure that they're measuring the clinical feature accurately.
Great. And maybe a follow-up question. How about a cell phone to record EKGs?
I think there's no problem with the use of a cell phone, provided the technology has been validated to make an accurate recording. So again, it all depends on the validation. That would tell us that, you know, the measurement made by a cell phone is equivalent to a measurement that might be made with a traditional machine. And we also have encouraged the opportunity to use, for patients to be able to use their own devices, to bring their own devices, provided the devices sort of satisfy the same specifications and can do the same thing. So yes, cell phones are probably going to play increasing roles in decentralized, or at least digital health technology measurements. Thanks.
And one more in this category, what is the FDA stance on the use of, quote, bring your own device, for instance, cell phones, for the ECOA PRO data collection? There is quite a bit of evidence on the ability for this modality to collect patient data accurately. How much more data would be needed for the FDA to support this? Experience of myself and colleagues' experience indicate that there's some reluctance.
So again, I touched on that in the previous answer, but I think FDA is definitely interested in facilitating that. I think we all understand that provided sort of devices that are owned by study participants may be very effective in capturing the data if they all satisfy the minimal specifications depending on what's needed. So for general things like patient-reported outcomes and so on, where the demands on the technology are limited, it's very likely that bringing your own devices will be satisfactory, but obviously that would have to be reviewed. For more specialized kind of measurements, like things like pulse oximetry or glucose measurements, I think there'd probably be a little bit more circumspection about whether bringing your own device is a good idea. We would have to make sure that all the devices provided equally reliable information. But I think we all understand that nobody wants to run around carrying dozens of cell phones in their pockets or being strapped to 6 smartwatches. So I think in the interest of convenience, it's important to try and facilitate the fact that patients are able to use their own devices, which they're familiar with and comfortable operating. Thanks.
Great, thank you, Leonard. John, a couple of questions for you here as well. The first, I was taught that observational studies are inferior to clinical trials. So why should we even bother with real-world evidence?
Thanks, Kim, and thanks for the question. I think the question refers likely to hierarchies of study design, which I mentioned in the presentation. They gained traction as of the 1990s. Unfortunately, when applied rigidly, those design hierarchies oversimplify the question of whether any particular study does generate valid evidence. We all agree that randomization is wonderful, and I'll add that the prospective research infrastructure that accompanies a trial is another underappreciated strength. But on the other hand, small sample sizes and other issues can and sometimes does threaten the validity of trial results. If we switch gears, I could say conversely, at the start, bias and confounding are indeed threats, serious threats to non-interventional studies, but such studies can be valid. As I mentioned in the presentation, we use a 3-pronged approach to get started. First, are the data fit for use, sometimes called fit for purpose? Is the study design adequate, i.e. rigorous? And last but not least, third, whether the study conduct is appropriate and adheres to our FDA regulations. As an aside, as was mentioned in the example of tacrolimus Prograf, when the anticipated effect size based on available knowledge suggests that the drug effect is large, that helps us to believe that the drug outcome association is real, even if we can't identify and account for every particular type of bias. So the bottom line is we still need clinical trials, make no mistake, but we shouldn't summarily dismiss observational, aka non-interventional studies. Thank you.
Thanks, John. And one more for you while you're here. In what way can randomized trials involve real-world data?
Thanks again. I did mention it perhaps briefly in the talk. As one example, electronic health records as a real-world data source can be used when appropriate, say to obtain information on the primary outcome in a point-of-care randomized trial. Both I and Leonard Sachs mentioned that particular type of study design. And I should add, in addition, most trials, if not all, already sometimes use data from EHRs, albeit in a modest way, say to incorporate laboratory values or other information from the quote-unquote bedside. But that practice, sometimes called the swivel chair activity, is where someone transfers, on the trial staff, transfers information from the records to the case report form, but that sort of flies off below the radar screen. So what we're talking about when randomized trials generate real-world evidence is more than, say, identifying patients for a trial or identifying sites or capturing their CBC, their complete blood count, or their chemistries, but rather have a fundamental impact, such as collecting the outcome for a trial. In that case, including our congressionally mandated need to report back to Congress how real-world evidence is being used in drug development. We're keeping tabs on both non-interventional, that is, observational studies, as well as clinical trials, sometimes generating real-world evidence. So I'll stop there, Kim. Thank you.
Great. Thanks, John. Fred, a couple more questions for you. The first is, I did not understand the interest in evaluating the intent to treat or ITT population. Why evaluate participants that did not receive treatment?
The reason why you want to evaluate patients who did not receive treatment is when you randomize a patient, you need the results of all the patients that were randomized. For those who did not receive treatment, there might have been a reason why they did not receive it. They might have dropped out of the study. They might have been intolerant to the patient, to them when they took the drug or study drug. There might have been a number of reasons, but to preserve the integrity of the randomized process, you want to not potentially bias the patient population by beginning to exclude patients that might cause a bias. So that is why we're interested in the ITT population. We've modified that a little bit. We tolerate the modified intention to treat by all patients who received at least one dose of drug. And then you could ask the same question. If they only received one dose, that might not be effective at all. But again, we're doing our best to preserve the integrity of the randomization process. If you go to the other extreme, just those patients who participated in the trial fully, those who did not violate the protocol, where you don't know why people violated the protocol, there could have been safety concerns or lack of efficacy, then you have a restricted population that is fully biased, that is independent of the randomization procedure that you had wanted to install in the first place. So that is why we are very interested in an ITT population. The minute you randomize them to some arm, you stick to it.
Great. Thank you, Fred. Leonard, another question for you, this time on decentralized trials. How is the FDA adapting its regulatory framework to ensure the integrity and reliability of data collected through decentralized trials, especially considering the diverse range of digital health technologies and remote monitoring tools used in these studies?
There are no specific changes to our regulations, at least at this time. A lot of these features have already been part of trials for many years, as I mentioned in my presentation. A lot of decentralized activities have taken place before. Some of the digital health technologies are also no different from technologies that have been used in traditional trials, things like ECG machines and blood pressure machines and glucose machines and so on. So technologically, I don't think we're in a completely new universe. There are a couple of considerations for data where we've expanded on some of the regulations. Many of the folks on the call may be familiar with our Part 11 regulations, and we have a guidance published, and now it's being revised and will be issued in final pretty soon on the considerations for data integrity and reliability as far as the electronic processing of the data goes. I think that's a novelty. Again, I don't think that there are any other fundamental differences between whether something's done locally or remotely, provided the data are supervised and the investigator knows what's going on. So the short answer is there are no new regulations, but obviously we've had to interpret older regulations in the context of these more novel trial designs.
Great. Maybe one additional follow-up question there that's related. In a decentralized trial, what sort of training is necessary for local healthcare providers?
I'm glad you asked that question, because our vision of local healthcare providers is that they should not need training. The reason we're interested in including local healthcare providers in trials is that we're using them for what they normally do. And they should not need to have a knowledge of the trial protocol or the investigational product. For example, if you're getting a local healthcare investigator close to a patient's home to do an ECG or to do a physical examination, that would be agnostic to the fact that the patient was on a particular investigational drug. For all those procedures that require knowledge of the protocol, for example, study-specific assessments or scoring of outcomes and so on, that should be done by study staff. We do not envisage local healthcare providers having to do that. That would require training, obviously. So the short answer is that there should not, based on the tasks that are allocated to local healthcare providers, training should not be required, although there's a possibility they may be given limited instructions, like have a patient sit for 5 minutes before you measure the blood pressure, but no specific training. Thanks.
Great, and thank you all. We have a lot of excellent questions and really appreciate all of your input on this session and the work you did to prepare for it. With that, I believe we are up to our first break and we will resume at 1:30 p.m. Eastern Time. Thank you all.
Welcome back from the break. We hope you're able to get a quick lunch to help move us right into our next presentation on trial design considerations in rare diseases by Dr. Scott Winicki, lead physician of Rare Diseases Team in the Division of Rare Diseases and Medical Genetics, Office of Rare Diseases, Pediatrics, Urologic, and Reproductive Medicine, and O&D. And the next presentation will be on special populations in clinical trials by Dr. Leonard Yao, Director in the Division of Pediatric and Maternal Health in the Office of Rare Diseases, Pediatrics, Urologic, and Reproductive Medicine, O&D. This will take us into our next Q&A session, moderated by Dr. Leonard Sachs. Please join me to welcome our first presenter, Dr. Winicki.
Thank you for that kind introduction. I'm Scott Winicki, and today we're going to talk about trial design considerations in rare diseases. For this talk, we have 4 learning objectives. 1, to discuss rare diseases and orphan product approval trends. Next, to discuss challenges and considerations in rare disease drug development, because development of drugs in rare diseases is quite difficult for a number of special considerations. I'm going to talk about resources that can assist those involved in rare disease drug development. And finally, I want to talk about 2 programs, CDER's Accelerating Rare Disease Cures Program, or ARC program, and the Rare Disease Endpoint Advancement Program, which I'll call RDEA, which is part of CDER's Prescription Drug User Fee Act 7 commitments. If we look at novel drug approvals for CDER from 2015 to 2022, we see that half, in fact, are for non-rare indications, and exactly half are for rare indications. So there's a lot of activity and approvals going on in rare diseases. And if we look back 40 years at the passage of the Orphan Drug Act, since 1983, FDA has approved over 550 unique drugs and biologics for over 1,100 rare disease indications. However, we know that 30 million Americans live with a rare disease, and that the vast majority do not have approved treatments. On this graph, if we look, the green bars are orphan novel drug approvals, that is to say for rare diseases or rare indications, and the non-orphans are in blue. And the purple line with the percentages shows the percentage of novel drug approvals which are rare. And you can see, looking from 2010 to 2022, that trend is steadily increasing. And in 4 of the last 5 years, more than 50% of drug approvals have been for orphan indications. Now I want to talk a little bit about expedited programs. These are programs that FDA has to help move drug programs through to approval more efficiently, so that after all, the goal here is to get approved drugs to the patients, right, to treat rare diseases with unmet medical needs. So there are 4 programs here. The first is Fast Track, then Breakthrough, Accelerated Approval, and Priority Review. Each of these has different qualifying criteria, and as long as you meet the qualifying criteria, you can participate in more than one of these. So I'm first going to talk about the qualifying criteria, then sort of what does that program get you? What are the benefits of being part of that program? So in Fast Track, it's for a drug that treats a serious condition, and that the non-clinical or clinical data demonstrate the potential to address an unmet medical need, which of course in rare diseases is something that we see commonly. For Breakthrough Therapy, also it's for a drug that treats a serious condition, and there's some preliminary clinical evidence that indicates the drug may demonstrate an improvement on a clinically significant endpoint over available therapies. Accelerated Approval, still for a drug that treats a serious condition, but provides a meaningful advantage over available therapies, and demonstrates an effect on a surrogate endpoint that is reasonably likely to predict clinical benefit, or on a clinical endpoint that can be measured earlier than irreversible morbidity or mortality. And finally, Priority Review is for an application for a drug that treats a serious condition, and if approved, would provide significant improvement in safety or effectiveness. Now here are the features of the program. So with Fast Track, it's actions to expedite development and review, as well as rolling review. So it doesn't require that all of the application be submitted at the same time, but that it can be submitted in sections. For breakthrough therapy, there's intensive guidance on efficient drug development, as well as an organizational commitment from senior leadership, and also rolling review. For accelerated approval, it's approval based on the effect of a surrogate endpoint or an intermediate clinical endpoint that is reasonably likely to predict a drug's clinical benefit. And for priority review, it's a shorter clock for review of the marketing application, meaning FDA has 6 months to review the application instead of the standard 10. Now I want to look at the use of these expedited programs by drug applications. So looking at the orphan in red and the non-orphan in blue. And you can see for each of these, priority review, fast track, accelerated approval, and breakthrough, that the orphan drugs are taking advantage of these at a higher percentage than the non-orphans. And I think that's good. That means that people are aware of these programs, that drug developers are taking advantage of those, and that's a good thing. And then you can see to the right of the graph, 88% of orphan novel drug approvals took advantage of at least one of these expedited programs, and 73% took advantage of more than one of these. So again, I think that's a great thing, and if you're a drug developer and qualify for one of these programs, it's definitely something that you should look into, because I think it's something that can, again, expedite the development, which is all about, again, addressing the unmet medical need for patients. Rare disease drug development is hard, and it's hard for a number of specific reasons. First, the natural history is often poorly understood. Some of these diseases affect very few people. Perhaps nobody's looked at it in depth, and we may have a poor understanding or just a very incomplete understanding of the natural history. Of course, many of these diseases are progressive, serious, and life-limiting. Most lack approved therapies, which means there's urgent need, unmet medical need, and many have pediatric onset. Inherently, in a rare disease, you're dealing with a small population, which often restricts or makes study design challenging. There's phenotypic and genotypic diversity within a disorder, so that even if a person has the same exact gene mutation as another person, those people may show up with different signs and symptoms, or they may show up with different signs and symptoms at different points in the lifespan. So often these are challenging because not every patient shows up with the same problems in the same way at the same time. Development programs often lack a solid translational background, meaning things like non-clinical data and animal models may not exist or may not be well developed. Drug development tools like outcome measures and biomarkers are often lacking because these diseases are not well understood. Sometimes we simply don't have good biomarkers that can help us better understand the disease and better understand drug effects. And there's also lack of precedent, specifically clinically meaningful endpoints. It's not always obvious what to measure that is clinically meaningful in a rare disease. It's also sometimes not easy to measure that. So those present additional challenges to drug development. There are also some considerations about the environment of rare disease drug development that can make it challenging. Often these drugs are developed by smaller companies with less regulatory experience, so that can present some challenges. On the upside, there are often active patient stakeholder groups who are looking to navigate and participate in drug development. Some of these are very experienced, others less so, but I think an active and interested patient population is definitely a plus. We also have an academic community, very dedicated, but sometimes they have limited knowledge of the regulatory requirements or aspects of clinical trial development. So I think the bottom line here is we all must engage our stakeholders to enhance the support of an understanding and to gain alignment. Because after all, we're all in the same team in terms of we want to address these unmet medical needs. We want drugs out there to treat people with rare diseases who don't have current treatments or whose current treatments may be inadequate. Now I want to move on and talk about CDER's Accelerating Rare Disease Cures program. So the ARC program's vision is speeding and increasing the development of effective and safe treatment options, addressing the unmet needs of patients with rare diseases. And the mission is to drive scientific and regulatory innovation and engagement to accelerate the availability of treatments for patients with rare diseases. What I want you to take away from this diagram is that ARC is an umbrella program. It's not an office in and of itself, but we work with many different offices across CDER, the Office of New Drugs, the Office of the Center Director, the Office of Translational Sciences. I come from the Rare Disease Program, so that solid curve shape at the bottom, I like to think of us as the connective tissue that holds all of these things together and coordinates the work between all these different offices. We often work with different offices and divisions within CDER, like the Office of Medical Policy or the Office of Surveillance and Epidemiology. And we actually work across centers beyond CDER, most often with the Center for Biologics, but also with the Center for Devices and Radiologic Health, the Oncology Center for Excellence, and very frequently working with the Office of the Commissioner. So rare diseases is not something that is siloed, but is something that operates across various offices within CDER, and we often work with other groups beyond CDER. Now I want to talk about some of the resources that have been developed by ARC that are for rare disease and hopefully will provide assistance and make finding information easier. One thing we're trying to do is to make the ARC page on the FDA website a one-stop shop for rare disease drug development resources. When we talk to outside stakeholders, one of the complaints that we heard was that FDA resources are often hard to find. For instance, we have a guidance document page and you can search guidances, but if you don't know the title of the guidance you're looking for, sometimes it's difficult to find the specific guidance that you need. So we're trying to make the ARC website this one-stop shop. One of the things we have on there are recordings, both the slides and the audio recordings of conferences we've held. So this can be a library or a resource. If you have a particular need about rare disease drug development, you can find the conference that fits the topic you need and go back and watch that conference. Or maybe if you attended the conference and there was a particularly interesting talk, you can go back and rewatch it. So two of these from 2022, one was jointly between FDA and NIH about regulatory fitness in rare disease clinical trials. So this is a topic that comes up very often. And this was CDER's Rare Diseases team, as well as the National Center for Advancing Translational Sciences from NIH. And the focus was on academic investigators and those looking how to bridge the gap between academic investigation and the regulatory aspects of drug development. Also back in May of 22, we did a workshop with Duke Margolis, Translational Science and Drug Development, Surrogate Endpoints, Biomarkers, and more. So again, it brings up that important role of translational science and the development of surrogate endpoints in rare disease drug development. This year we had two additional conferences. First with the Johns Hopkins Center of Excellence in Regulatory Science and Innovation, the Johns Hopkins CIRSE. That was advancing challenges in the design and analysis of rare disease clinical trials, considerations and tools. And it talked about things like how to collect high quality and fit for purpose data and the use of real world data to inform rare disease drug development. And then we had another conference with the University of Maryland CIRSE about quantitative systems pharmacology. So a bit earlier I mentioned the guidances. So if you go to the ARC page on the FDA website, you will see this box. If you click on it, you'll see the landing page shown on the right. And what we've done is taken all of the guidances that are related to rare disease drug development that might be relevant, that we think our stakeholders would be looking for, and organize them by categories. So you can look simply under the wide-ranging category of rare disease, but also under clinical trials or real-world evidence or digital health. So we've sorted those by topic, so hopefully we've made those guidances easier to find. If maybe you don't know the specific guidance that you're looking for, this hopefully makes a resource that makes what you need easier. I want to highlight two recently issued guidance documents. One is Considerations for the Design and Conduct of Externally Controlled Trials for Drugs and Biologic Products. This came out in February of 2023. We often get questions about externally controlled trials. and under what circumstances can they be used, and this guidance can be a resource there. The next guidance came out just in September of 2023, demonstrating substantial evidence of effectiveness with one adequate and well-controlled clinical investigation and confirmatory evidence. So this is often a pathway that is used in rare disease drug development, and this sort of guidance can be useful to you if that's a pathway you're pursuing for your therapeutic. We've also compiled a list of funding pages, of funding opportunities for rare disease and product development research. So again, if you go to the website, click on that box on the right, you'll get the list of funding opportunities. We also have a quarterly newsletter. You can sign up for it, and we will email you quarterly everything that's happening in rare diseases, just sort of news highlights, so you can stay abreast of rare disease drug development news. Now I want to talk about one of our educational programs, Learning and Education to Advance and Empower Rare Disease Drug Developers, also called LEADER 3D. And as I mentioned earlier, we went out and sought input from stakeholders who design or conduct rare disease drug development programs to identify the knowledge gaps about the regulatory process and where we could perhaps develop or expand educational resources for stakeholders. So I already mentioned sort of that guidance list. One of the things we heard was guidances are hard to find. So we're trying to make that easier. And we're in the process of additional resources. Again, the idea being to better understand the challenges in bringing rare disease products to market, identify those knowledge gaps, and then produce educational materials on relevant topics. And when we talk to our stakeholders, both within and external to the federal government, we heard that non-clinical and clinical pharmacology was a big issue, clinical trial design and interpretation, and also regulatory considerations. I want to mention that in parallel with the LEADER 3D effort, the CDER Patient-Focused Drug Development, or PFDD group, is working with the National Organization for Rare Disorders, NORD, to develop an advanced drug development education series that's geared toward patients and patient groups. Our materials are more geared toward rare disease drug developers, but I think patient groups often have a keen interest in these same topics. Now I want to talk about the ARDEA pilot program, again, the Rare Disease Endpoint Advancement Program. So the idea here is it's a pilot program that seeks to advance rare disease drug development programs by providing a mechanism for sponsors to collaborate with FDA. And this would be throughout the efficacy endpoint development process. And the idea here is to, again, to increase the development of endpoints because they are often lacking for many rare diseases. So eligibility is that you should have an active IND or pre-IND, and that the proposed endpoint is a novel efficacy endpoint. This is really quite a new program. It started at the end of fiscal year 23. It's going to go through fiscal year 27, and we're going to accept up to one proposal per quarter or a maximum of 3 proposals per year between fiscal years 2024 and 2027. Now, there is a disclosure or transparency aspect of this. We're going to host up to 3 public workshops by the end of fiscal year 27 to discuss various topics related to endpoint development. And the novel endpoints that are developed through this program may be presented by FDA in things like a guidance document, a public-facing web page, or one of those public workshops that I mentioned. We already held one of those public workshops, Novel Endpoints for Rare Disease Drug Development, in conjunction with CDER, CBER, and Duke Margolis. We held it in June. Talked about the application process and required elements for ARDEA, but also scientific and technical issues that are associated with developing endpoints, and lessons learned from previous PDUFA meeting programs. This recording of this is also on the ARC website. So again, if you're interested in endpoint development, this would be a good resource. Here's the URL for the ARDEA update page. So there you can get FAQs, learn about meeting packages or disclosure elements, and other endpoint development resources. Now I want to touch on another development program, the Support for Clinical Trials Advancing Rare Disease Therapeutics, or START pilot program. And the idea here is to accelerate the pace of the development of certain either CBER or CDER regulated products that are intended to treat a rare disease. This is a product focused and designed to augment the currently available formal meeting structure between FDA and sponsors. It's milestone driven, so the idea is to take a program and get it to that next big step. Maybe that's getting it to be ready to start its pivotal clinical trial, or getting it to the pre-BLA or pre-NDA stage. It can address a variety of specific programmatic issues. I think it depends upon the particular product, but that could be clinical or non-clinical considerations. And for CDER, the focus here is on rare neurodegenerative conditions, including those of rare genetic metabolic etiology. CBER has slightly different qualification criteria. So this is really a brand new program. Applications haven't even opened yet. That'll start on January 2nd, 2024 and go to the beginning of March. And interested sponsors are asked to submit their request as an amendment to their IND. Each center, CBER and CDER, will pick up to 3 programs to be part of this pilot program. But in the future, we might expand this program, or there could be a second iteration of the program. So now I want to talk briefly about 2 case studies, sort of illustrating the challenges of rare disease drug development and how the resources can be helpful. So first we have Velminase, and the indication here is for the treatment of non-central nervous system manifestations of alpha manocytosis, or AM, in adult and pediatric patients, and it was approved in February of 2023. So a little bit about alpha manocytosis. It's a rare autosomal recessive lysosomal disorder. It's an enzyme deficiency, and that deficiency leads to the accumulation of oligosaccharides in various tissues, and involvement includes the central nervous system as well as the musculoskeletal and immune system. And Velminase, the drug, catabolizes these oligosaccharides that accumulate in the lysosomes. So again, just to remind you of the common challenges in drug development and why drug development in rare disease is hard. And I think that Velminase illustrates a number of these things, right? We talked about diseases being progressive, serious, and life-limiting. Well, AM is a serious disease that can lead to death in early childhood, or in later onset forms, it can lead to progressive and severe motor impairment. In terms of small populations, it have a prevalence of about 1 in 500,000. And it's highly variable in its clinical manifestations, severity, and progression. So obviously that makes conducting a clinical trial more challenging. In terms of drug development tools and outcome measures and biomarkers, the trial demonstrated a statistically significant reduction in serum oligosaccharides. However, that's not a validated surrogate endpoint, so it couldn't be the primary endpoint for the trial, but it does support the proposed mechanism of the drug. And in terms of clinically meaningful endpoints, several different ones were measured for the trial, but this included the 3-minute stair climb test, the 6-minute walk test, and the forced vital capacity % of predicted normal value. So what resources could be useful if you had a drug development program like Velminase? Well, it was approved based on one adequate and well-controlled trial plus confirmatory evidence. So we have the guidance list there, and we highlighted the one adequate and well-controlled investigation and confirmatory evidence guidance. Endpoint selection is key in clinical trials. So we have the Rare Disease Endpoint Advancement Program, and we have the workshop recording on our website talking about endpoint development. And perhaps if there was a need for additional meetings, the START program is something that, as a rare disease drug developer, you might want to take advantage of. Case study 2, vutrisiran, which was approved in June of 22 for the treatment of polyneuropathy of hereditary transthyretin-mediated amyloidosis in adults. So a little bit about hereditary transthyretin-mediated amyloidosis. It's an autosomal dominant disorder. It's caused by mutations in the TTR gene, which leads to protein misfolding, aggregation, and deposition in the nervous system, heart, kidneys, eyes, bone, and GI tract. So the drug's a small interfering RNA that targets the TTR mRNA, and at the indicated dose, vutrisiran reduced TTR levels by an average of 85%. So again, thinking about the challenges here, it's a serious and progressive disease, life-limiting. Death occurs within 5 to 12 years of onset. And even though there are other approved therapies at the time vutrisiran was approved, there was still significant unmet medical need because not all patients were able to receive or tolerate the currently available treatments. In terms of small populations, really quite rare. HATTR is estimated to be between 5,000 and 10,000 persons globally. And to mention the translational background, the reduction in serum TTR provided strong mechanistic support for this drug. So what about resources? Again, it was approved using that one adequate and well-controlled plus confirmatory evidence pathway. So we have that guidance. The clinical trial also used an external control. So again, to highlight the considerations for the design and conduct of externally controlled trials guidance. And then just to highlight two of the relevant workshops that we have, the recordings on the website, the FDI NIH Regulatory Fitness in Rare Disease Clinical Trials, and the Translational Science and Drug Development, Surrogate Endpoints, Biomarkers, and more. So in conclusion, I want to make a couple of points. In recent years, over 50% of CDER's novel drug approvals were for rare diseases. That CDER's ARC program will help CDER work more effectively with our rare disease drug development partners, and that CDER and ARC have multiple resources to assist with the challenges of rare disease drug development. I'd like to end with a couple of challenge questions. First, educational content on the ARC website includes A, a list of guidances organized by topic area, B, recordings of public workshops, C, listing of funding opportunities, or D, all of the above. And the answer is D, all of the above. Challenge question 2. Challenges in rare disease drug development include A, small populations, B, lack of precedent including clinically meaningful endpoints, C, phenotypic and genotypic diversity within a disorder, or D, all of the above. And the answer is D, all of the above. That concludes my presentation. Thank you for listening, and I'll be back shortly for the question and answer session. Thank you.
Hello, everyone. I'm delighted to be back for this year's FDA's Clinical Investigator Training course and to give you a few perspectives that I have on specific populations in clinical trials. This is my disclosure slide. In addition, I want to just point out a couple of things. The first is that my talk will focus on specific populations as defined by FDA, and specific populations is a newer term. You may have heard the older term special populations, but we use specific populations now because we think it's a more appropriate and probably more precise term. The second point I want to make today is that it's going to be a whirlwind talk, and I'm really just going to provide an overview and important guidances that FDA has published recently to provide additional, more in-depth information for you to consider. And then thirdly, that I am a pediatric nephrologist, so while my clinical expertise is limited to pediatrics and pediatric nephrology, I'm going to be talking about several specific populations today. So what do I hope to go over with you in the next 30 minutes or so? I want to review with you the inclusion of specific populations in drug development. And the three populations I really want to focus on today are children, pregnant and lactating individuals, and geriatric populations. In addition, I do want to also discuss enhancing overall diversity in clinical trials. So why am I giving you a talk on specific populations? Let me just say that on this slide, I think it demonstrates the importance that FDA has on collection of information on specific populations. Under the physician labeling rule, which is a regulation that describes, importantly, the content and format of prescription drug and biologic product labeling, there are specific sections under Section 8 that describe important information that should be included in specific populations, and you can see sections 8.1 through 8.5 listed here. I'm going to go over, each of these populations during my talk today, and let me just also note that there are other specific populations that I will not be discussing, and those include specific populations that have hepatic and renal impairment. I'm going to focus today mostly on pregnancy, lactation, pediatrics, and geriatrics. So the first topic I want to review this morning is related to pediatric patients and the inclusion of pediatric patients in clinical trials. So this slide really goes over two basic tenets that are foundational to pediatric therapeutics development. First, that pediatric patients, just like any population, including adults, should have access to medicines that have been appropriately evaluated. And what flows from that is that if a product development program includes adults, but children are likely to use the drug once it's approved, that development program should also include pediatric studies. In the next few slides, I'm going to go over some basic ideas and basic principles that underlie therapeutics development in children. The first of those are the ethical and feasibility considerations. There was a guidance that was just published a couple months ago in September of 2022 on the ethical considerations of clinical investigations of medicinal products or medical products involving children. Among other really important concepts brought forth in this guideline are the three basic tenets and three basic principles that you see here. First, that children should only be enrolled in clinical trials if the scientific objectives cannot be met through enrollment of subjects who actually can provide informed consent personally, such as adults. And because children are a vulnerable population, because they cannot provide informed consent, then clinical trials involving children must offer the prospect of direct therapeutic benefit. And without that prospect of direct therapeutic benefit, the risk to the child for enrollment in a trial should be low, and low is basically defined as no more than a minor increase over minimal risk. And those notions and concepts of minimal risk and minor increase over minimal risk are really well defined and described in this guidance. And then finally, the children really should not be placed at a disadvantage by being enrolled in a clinical trial. But even when these when these ethical considerations have been incorporated, there are also feasibility issues and feasibility considerations that are commonly seen in pediatric therapeutics development that are not necessarily as commonly seen in adult product development. And this is, again, a good thing for children because the prevalence or incidence of many diseases and conditions are lower when compared to adult populations. In this next slide here, I want to go over some important concepts that are brought forward in this ICH E11R1 addendum document that I had mentioned a couple of slides ago related to the foundational principles. And in this important addendum guideline that was published by FDA in 2018, there are several areas related to pediatric therapeutics development that are found in this guidance. And it's an excellent guidance to those of you who are interested in learning more about how to conduct pediatric drug development programs. On these three bullets you see next to the guideline, I think I want to highlight these three areas. The first is the use of a strategy called pediatric extrapolation This can be used to increase the efficiency and feasibility of pediatric product development. And the basic idea behind pediatric extrapolation is that when there are similarities between a pediatric population and a reference population, usually an adult population, that there are similarities in the course of a disease and the response to whatever drug you're studying, that those similarities can be translated into leveraging information that could lead to clinical development that requires less than the standard adequate and well-controlled trial. And this guidance does give some high-level descriptions of pediatric extrapolation. In addition, there is really clear guidance about the enrollment of children stratified or staggered by age. You know, typically we see, and historically we've seen adolescents studied first, then school-age children, then preschool children, then toddlers, then infants, and then newborns. And the guidance puts forth the important consideration that strategies that are relying on staggered enrollment based solely on age really should be justified, because if there's really no reason to study adolescents only first, there's no safety or efficacy reason to not enroll younger populations at the same time, that one should consider that really, because it decreases the time it takes to have the drug studied and therefore then potentially available for children to use. And then finally, the guidance goes over many other unique product development considerations, including age-appropriate formulations and safety information, including the need often for long-term safety information if the long-term safety is unknown in a pediatric population. I want to highlight on this slide one recent guidance that was published again in 2019 about actually including adolescent patients in adult trials. And here the Oncology Center of Excellence published this guidance about inclusion of adolescent patients in adult oncology trials. And really just to suffice to say that there are certain situations in which adolescent oncology patients are very similar, in fact, to adult oncology patients, including young adult oncology patients. And in those situations, adolescent enrollment should be considered to really, again, speed the availability and not delay access of potentially effective therapies to the adolescent oncology patient. And you can see in the second bullet some considerations when it would be appropriate to consider inclusion of adolescents in adult oncology trials. So taken together, I'm just giving you some highlights about considerations related to inclusion of pediatric patients in clinical trials, and just some recent guidance that FDA has published. So moving on, I want to spend a few minutes to talk about the inclusion of pregnant individuals in clinical trials. And as a background, again, no different than children, pregnant individuals should have access to medicines that have been appropriately evaluated. Now, one important distinction between pregnant individuals and children are that medicines for specific indications that are approved in adults are also approved in pregnant adults unless specifically contraindicated. In other words, a pregnant adult is still an adult, and so when the drug has been tested and approved in adults, then one does not need to say that a clinical trial needs to be conducted in pregnant adults to ensure that the efficacy is similar, unlike the situation in children. However, pregnant individuals are often automatically excluded from clinical trials. And we also know that alterations in physiology during pregnancy can affect both dosing and safety in the pregnant individual, that because pregnant individuals are often automatically excluded from clinical trials, there's often little to no human data to support the safety and dosing of the product prior to approval. And that leads to prescribers and pregnant patients really left to make benefit risk decisions without any data. And that situation is really unacceptable. And so recently, FDA has been working with external stakeholders and drug developers to consider what types of information are important to obtain to support, the appropriate dosing and safety of drugs when they're used during pregnancy. One important initiative that was undertaken by FDA over the last 7 years is the Pregnancy and Lactation Labeling Rule, or PLLR. And the reason I bring this up is not to really go into, the changes that occurred in pregnancy and lactation labeling under PLLR. And you can see in this slide, there was a shift in some of the location of the information. But more importantly, the PLLR was intended to improve the communication of risk information that was collected as part of the labeling of a product. But in the implementation of PLLR, FDA learned quite a bit about the state of data availability in drugs used during pregnancy. And in many cases, even though the framework and the new labeling framework was successful in improving the communication of risk when there were data that supported a specific risk, oftentimes what we learned was that data were actually absent, and that even though there was the ability to communicate in a better way, there were no data to communicate. And even when there was data available, often it lacked both quality and quantity. Finally, although there was a great desire to include definitive risk statements in labeling, often there weren't sufficient data, and that's the current state, that there are oftentimes insufficient data to really be definitive about the risks of the drugs when used during pregnancy. So as a consequence to what we learned from the implementation of PLLR, FDA published a guidance on the inclusion of pregnant patients in clinical trials. And this guidance, this draft guidance published in 2018, includes situations when pregnant individuals, pregnant women can be included in clinical trials. It describes and follows the HHS framework for human subject protections. And it really does consider, the inclusion of pregnant individuals both in the post-market and pre-market setting. And finally, an important consideration of this guidance also is what to do when a patient becomes pregnant during a clinical trial. So all of these important considerations are described in this guidance, and I do think that it provides really a roadmap and perhaps a call to consider when pregnant individuals can be safely and ethically enrolled in clinical trials. And let me just say here too that we're not proposing that pregnant individuals just automatically be included in clinical trials, because there needs to be important safety information collected before including pregnant individuals. However, I think what this guidance put forward is the idea that automatic exclusion of pregnant individuals in clinical research is also unacceptable. And there has to be a careful review and justification for why pregnant individuals would be excluded. In addition, I think this guidance also sets up important considerations about early discussions and early planning so that the appropriate reproductive toxicity studies can be completed in non-clinical studies so that when the phase 3 trial is ready to begin enrollment, there is already some information that can help guide the safety and the safety monitoring of a drug to allow for pregnant patients to be enrolled in clinical trials. So I just want to spend a few minutes about the collection of important safety information on when drugs are used during lactation. And here again, the problem statement is similar to the problem statements I've already provided you on research in children and pregnant individuals, that medications are used in lactating individuals, often with little to no human data about the safety of the drug, in the breastfed infant. And importantly, collection of data on the transfer of drug into human milk and into the breastfed infant can provide really important information that can help prescribers and patients. Unfortunately, such data are really limited in terms of availability at this time, and generally, almost never do we have the information before the drug is approved. So in 2019, FDA published a guidance on clinical lactation studies. And just briefly here, I want to highlight some of the important considerations in this guidance, including describing scenarios when a lactation study would be appropriate. And also, if you're planning to conduct a lactation study, discusses ethical considerations about who can participate in lactation studies, including lactating women who are already taking the drug for medical or prescription or therapeutic purposes, and also potentially the inclusion of healthy lactating women in lactation studies as well. And those ethical considerations are described in this guidance. There are really, I think, well-described sections about different study designs that can be used and the information that can be obtained in such study designs. And then finally, sampling strategies and pharmacokinetic analysis methods are described. So this is a really, I think, complete guidance for those interested in getting more information on how to and when to conduct clinical lactation studies. So now I want to talk about the last specific population, the inclusion of geriatric patients in clinical trials. FDA has defined as part of their guidance on geriatric information in prescription product labeling as patients 65 years of age and older. And again, here, like pregnant individuals, medicines approved for indication adults are also approved in geriatric populations unless specifically contraindicated. Now, it's important to consider the inclusion of geriatric patients for many reasons, and certainly the three that I've highlighted here on this slide are major considerations. That comorbidities that occur more commonly in geriatric patients could have effects on the efficacy and safety of the product. For example, organ dysfunction can play a greater role in geriatric patients because more geriatric patients, for example, may have organ dysfunction. There is certainly a higher incidence of concomitant therapy and therefore a higher risk for drug interactions. And simply that the geriatric population is increasing in the United States, and therefore, in order to appropriately study drugs and patients are going to use them, if there are more geriatric patients in the United States, then there are likely going to be more geriatric patients taking any given medication. So, importantly, the labeling guidance describes when geriatric information should be placed in that section 8.5. And the labeling should provide information, if any are collected, on differences in the safety, effectiveness, or dosing, that is pharmacodynamics and or pharmacokinetics, in a geriatric population compared to a non-geriatric adult population. Here again, the Oncology Center of Excellence leads the way and has recently published a guidance in March of this year on the inclusion of older adults in cancer clinical trials. Again, outlining why it's important to study geriatric patients in oncology trials. And you can see the at least three reasons here. The guidance also goes over ways to incorporate geriatric patients in trial designs, and also provides approaches to improve recruitment and retention of geriatric patients, and also gives additional information on important and specific adverse event monitoring and management in geriatric patients, and the potential need for collection of additional post-marketing data when geriatric patients are enrolled in clinical trials. But here, really all intended to improve the generalizability of cancer treatments to include all the populations who might benefit from its use, including the geriatric population. I want to spend the last few minutes talking about another important topic that's not specifically related to specific patient populations, but really overall diversity and enhancing diversity in clinical trials.
I think our FDA Commissioner, Dr. Robert Califf, said it best when he said, quote, "The U.S. population has become increasingly diverse, and ensuring meaningful representation of racial and ethnic minorities in clinical trials for regulated medical products is fundamental to public health." I think FDA is committed to ensuring the health of the entire U.S. population, and many patients in this country continue to be underrepresented, both in drug development and in access to medicines. FDA is currently working in many areas to address this underrepresentation. For example, FDA just recently convened a public meeting on November 29th and 30th on discussing approaches to enhance clinical study diversity. This meeting was convened in collaboration with the Clinical Trials Transformation Initiative, or CITI. And the goal of the meeting was to solicit input on, first, increasing enrollment in clinical trials of historically underrepresented populations, and secondly, how we can encourage clinical study participation that actually reflects the prevalence of the disease among demographic subgroups. There should be recordings available as well as additional publications as a result of this public meeting. So FDA has put out two recent guidances that describe enhancing diversity in clinical trial populations. Their first guidance, published in 2020, includes strategies to broaden eligibility criteria and also provides guidance on inclusive trial practices. And you can see here many topics discussed in the guideline, the guidance that go over ways to enhance diversity, from clinical trial design and methodologic approaches to enrichment strategies, and then importantly, decreasing burden and enhancing retention. Both are important, not only to get patients enrolled, but also to keep them in the trial once they are enrolled. And there is finally a section on rare disease considerations. So again, a really important guidance that describes how clinical trials can be potentially more diverse and inclusive of populations who are likely going to be taking the drug. This slide goes over, I think, an important development in improving diversity as part of drug development, drug and device development. And it is a joint guidance sponsored by no less than 5 centers within FDA. And it discusses the inclusion of formal diversity plans as part of drug development. And you can see here that in the opening of an IND or IDE, that a diversity plan is recommended when studies that are being conducted under the IND or IDE are intended to support a marketing application or device marketing application. Diversity plans should be submitted early so that they can be discussed with the agency and advice given no later generally than end of phase 2. And also provides detailed description of what should be included in the diversity plan. I'm not going to go over each of the items here. It's really beyond the time that I have, but I have listed them here so that you can get a flavor for what types of information should be included in a diversity plan. I really think that the inclusion and the recommendation to include diversity plans in all IND or IDs that are intended to move to a marketing application will really move forward the inclusion of appropriate and diverse populations in drug development. So finally, I want to briefly mention the role of decentralized clinical trials in enhancing diversity in product development. A decentralized clinical trial is really just a clinical trial where some or all of the trial-related activities can occur at locations separate from the central or principal investigator's location. You can see here several potential benefits that can enhance diversity, including avoiding having to travel long distances or take off work or expend additional cost to participate in a clinical trial. Those certainly you can imagine would enhance diversity, particularly in populations where it is difficult to participate because of socioeconomic reasons. In addition, it can also help for conditions that are rare or where the diseases are sporadic, so that those assessments can be made very close to the time and the place where some of the symptoms are occurring. Now, of course, importantly, there are challenges related to decentralized clinical trials, including the need for more staff, because you're just going to have a collection of data at more and disparate sites, and that can lead to compliance issues as well. So there needs to be really good documentation, really good training to make sure that every site in a decentralized clinical trial is collecting information in the same way, and that the data that can be used are valid and reliable. Nevertheless, again, I can't overemphasize, I think, the potential utility of a decentralized clinical trial in enhancing diversity. So I hope to have provided you a high-level summary of the inclusion of specific populations in clinical trials for drug development. And on this summary slide, I have 4 takeaways that I think are important for you to remember. The first is that pregnant and lactating and children and geriatric populations all should have access to medicines with information to support their safe and effective use. Secondly, that drug development should be designed to include specific populations when use is expected in these populations. And certainly there are unique ethical considerations in both clinical trials for children as well as pregnant individuals. Early planning is the key to making sure these specific populations are included appropriately. And remember that the U.S. population is really becoming increasingly diverse. And it's really important that we all consider how to be inclusive in clinical trials so that the clinical trials and the data that are obtained from those clinical trials can be generalized to reflect the population that is most likely to use the drug. And finally, the challenge question. Specific populations that should be considered for enrollment in clinical trials during drug development include A, pediatric populations, B, pregnant and lactating populations, C, geriatric populations, D, all of the above, or E, A, and B only. And hopefully you've all gotten the correct answer, which is D, all of the above. I want to thank you for your attention, and I look forward to the question and answer period at the end of this session.
Well, thank you very much, Scott and Lynn, for excellent presentations. So just to remind ourselves where we are, in the morning we spent some time discussing the overall design of clinical trials in general and some of the new initiatives that are underway to increase the reach of clinical trials, including technology and real-world evidence. And now during this session, we pivoted to looking at particularly challenging areas of drug development, specifically rare diseases and diseases that affect specific populations. So I think let's turn to the questions now. And I'm going to start with a question for Lynn here. Lynn, what do you think are the most important barriers to identifying specific populations who can be enrolled in clinical trials?
Thank you, Leonard. Really excellent question. And I think the most important thing to consider in identifying barriers, pediatric, pregnant, geriatric populations, is to really understand the population who has the disease, and then to very early in development, come up with a plan that addresses that. And I think that, we say all the time in our shop that there's no replacement for early planning. And so I think a lot of the troubles and a lot of the barriers can be overcome by having early conversations with FDA and really working on a plan that includes all these patients early in development. Thanks.
Thanks very much. And a question for Scott here. I think this is something that probably would be good to kind of expand on. The question is, how is CEDA's ARC program different from a rare orphan disease designation? And the questioner goes on to ask, does CEDA's ARC program affect accelerated approvals in rare orphan diseases? So maybe you can tackle or disentangle the concepts for us, Scott.
Thank you, Leonard. So the ARC program, completely different from the Orphan Disease Designation Program. That's something that's done by the Office of Orphan Product Development and the Office of Commissioner. The ARC program is a CEDAR program, and we're not an expedited program, nor are we directly involved in regulatory review. The program's role is to drive the development and the use of regulatory and scientific tools to support rare disease drug development and regulatory science. So very different than the Orphan Disease Designation Program, and not involved in regulatory review, so does not directly impact accelerated approval. Thank you for the question.
Thanks very much, Scott. I'm going to pivot back to Lynn. Lynn, here's a question, which is certainly something I often ask myself. Can a phase 2 study with pregnant women be conducted ethically? I think the person who's asking the question is obviously aware of the risks to the fetus, to the pregnant woman, and the sort of background safety data that may be available. So I guess, what's your view on conducting phase 2 studies in pregnant women?
Wow, Leonard, that's a complicated question, but a really good one. Let me just say that unlike the pediatric population that I described as a vulnerable population, because again, children are young such that they cannot provide their own independent consent. They can provide assent, but we really need to make sure that when we enroll children in clinical trials, that we get parental permission, that those informed consent documents are signed by guardians, and that children, if available, can provide their own assent. However, in the setting of pregnant individuals, they are not a vulnerable population, okay? They can make their own decisions, and they are able to provide their own informed consent. So the reason that pregnant individuals could potentially be enrolled in phase 2 PK studies is that if we have the appropriate safety information in place, and that is usually in the form of the important repro-tox studies, the pre and postnatal development studies that should be conducted. Oftentimes those aren't conducted very early in development, and that really precludes enrollment of pregnant individuals in phase 2 or phase 3 trials. So the answer is that it's not so much an ethical issue as it is a safety issue, because pregnant individuals can provide informed consent, they can make decisions, and we just need to make sure that the safety information is available to decide whether or not the timing is appropriate to enroll those pregnant individuals either in phase 2 or phase 3. I hope that helps to answer the question. Thank you for that.
I think that certainly gives us a good view of this, a good strategy. I turn back to Scott. I have a question here from somebody who mentions, I've noted that sickle cell disease has been characterized as a rare disease, although it has a high prevalence in ex-US, outside the US. They mentioned sub-Saharan Africa. is it the US prevalence that determines rare disease status by FDA, or the other things that determine whether it's regarded as a rare disease? Scott, do you want to tackle that? It's a technical question.
Sure, I'll tackle that. Thank you, Leonard. So rare disease, the legal definition in the United States is fewer than 200,000 persons with the disease in the United States. So it doesn't look at the frequency or the prevalence of the disease ex-US, only the US population, and the number is less than 200,000. So good question, and it points out that it is a very specific definition, and the worldwide prevalence doesn't play into it. Thanks for the question.
Great, thanks. I'm going to swing back to Lynn. There's a question here about one of our attendants will be working on a pediatric trial during the next year, and this individual mentions that there's already an FDA-approved drug on the market for the disease in question. What do you suggest to get parent buy-in for their children to participate in a clinical trial when there's already something available? They anticipate challenging enrollment. Any thoughts about helping them to enroll patients?
Yeah, and thank you, Leonard. Another really insightful question, and one that we have faced often in therapeutics development. I didn't provide this statistic in my talk, but on average, it takes somewhere for between 7 and 9 years after adult approval for the pediatric approval of that same drug. That's 7 to 9 years of off-label use, and during that period, and when there is off-label use, practitioners, patients, they have decided on their own and at their own risk that a drug works or doesn't work for them. So it's particularly important that we get trials started in children, as follow-on for adult drug development as quickly as possible, and to finish those studies as soon as possible to avoid and minimize that period of off-label use. So the question is, how do we do that? Particularly when there's other options available. And I would argue in many cases, the option of the unapproved drug that was already approved for adults is also already available because it's being marketed. And so I would say that, it requires a lot of effort on, in the ecosystem, I would say, to educate and to reassure and to change culture. And some of that has to do, I think, John Concato talked about it a bit. Leonard, you did too in the talks this morning about how to get the trials to the patients. And in addition to doing that, it's how do you get the buy-in and the trust in the community in whom you're going to do the trials. And that requires, I think, more than just, well, we got a drug, let's set it up and run the trial and then break down the trial infrastructure and then set it up again. So there needs to be a commitment to developing trust within the communities where the drug is going to be studied. That's not easy. I'm not suggesting that it is easy. But, developing those relationships and use of, pediatric clinical trial networks can definitely help to do that. I think also in figuring out how to get the trust or how to get the buy-in of the parents, it's also, we have lots now. I can describe 2 pediatric patient networks. One is the International Children's Advisory Network, or ICANN, in the United States. And then there's another organization in Europe called EYPAGNet, which is a European, I'm going to forget the exact acronym. But basically, these are organizations led by children who help other children understand the importance of understanding their disease and even potentially enrolling in clinical trials. So I think getting buy-in of parents involves getting buy-in in the community as well as getting buy-in from the children themselves. So maybe a sort of roundabout answer, but I hope that helps the questioner. Thanks.
Oh, I think those were actually very good answers. And while you were speaking, it occurred to me that this is pretty much a problem that applies to Scott's area as well, which is rare diseases, where I'm at least aware of certain situations where patients with rare diseases are using off-label treatments, and obviously one wants to get in with a trial before that becomes the norm. I don't know if you have anything to say about that, Scott, before I go into the next question.
Thank you, Leonard. Not particularly. I think just that it is always challenging in the rare disease space because we're dealing with many fewer patients. So it's not as easy, and I think it just emphasizes the point that probably everybody speaking today has made, and that is that the design of a clinical trial is crucial and needs to be tailored toward the situation, the disease, the patient population that it's focused on. Thanks for that question.
Yeah, I think that's a good answer. So while I have you on the line here, a couple more questions which are more of administrative nature. One is, can the priority review designation be granted by FDA before or after NDA submission? Any thoughts on that?
Certainly, Leonard. So priority review, and that's the expedited program that shortens the review clock from the typical 10 months to 6 months. You can apply for that with submission of the NDA or BLA, and the FDA responds to that request within 60 days. Thanks for the question.
And just to follow up on that, there's another question that's somewhat related. What is the timeline relationship between accelerated approval and priority review? I think maybe it's worth just outlining what the differences are between those programs. Any thoughts there?
Certainly. So accelerated approval is related to the use of a surrogate endpoint. And in terms of timeline, this should be discussed with the review division early, because I think the use of a surrogate endpoint is often an important issue in the review. So earlier there is better. And then I would say for priority, again, as I just stated a moment ago, that's with the submission of the NDA or BLA and shortens the review clock rather than, again, accelerated approval focused on what to use as an endpoint. Priority review shortens the review clock from 10 months to 6 months. I should also say that in terms of some of these details, because I know I went through the 4 expedited programs rather quickly, and they each have somewhat similar but different qualifying criteria. Likewise, they have different benefits. There's a guidance on this. And just to bring it back around to my talk, we have that guidance page. So if you go to the ARC website, just search FDA ARC, you should get our webpage. If you go down to that little guidance block, you can look on expedited programs. So the title of the guidance is Expedited Programs for Serious Conditions, Drugs and Biologics. And that guidance has all of the information I think that you would want to know about each of those 4 expedited programs. Thank you.
Thanks very much, Scott. That's very clear. I think we've got time for one more question for Lynn. Lynn, what do you think about this one? For children pregnant and lactating, do you think what do you think of trial-specific phase recommendations? And they talk about first in human. Any thoughts about that?
Yeah, that's a good one, Leonard, an interesting one. Hopefully I can answer within the time remaining. I think what I would say generally is that we have seen absolutely, and Scott can describe this too, in the rare disease setting, that yes, in fact, you can go first in human, even in children, particularly if there's really no correlate of disease in adults. In fact, you might want to wait until the adult program is completed and there's actually post-marketing safety data in adults before you even enroll a single trial, a single patient in a pediatric trial. And so what that speaks to is that the decision to move forward in a pediatric trial is very context specific, and that there is no absolute prohibition to first in human or waiting until the post-approval and long-term safety is already collected. And it really depends on all of the factors that I think we've heard throughout the day already, both in regular drug development, as you heard Fred give the talks this morning, all the way through Scott's discussion on rare diseases. So I would say that there is no absolute, prohibition to first in human, just as there is no reason necessarily to wait until after phase 3 is completed, the drug is approved in adults. Thanks.
Great. Thanks for a very thoughtful answer to a challenging question. I think that brings us to the end of our time. I wanted to thank both Scott and Lynn for a fantastic session on some very important areas of challenging areas of drug development. I also wanted to thank the audience for great questions. And at this point, we'll adjourn for our 15-minute break. Thank you.
Welcome back from the break. We're on the home stretch now, right into our first presentation in this final session on safety considerations in clinical drug development by Dr. Shabnam Nasir, Medical Team Leader, Division of Anti-Infectives, Office of Infectious Disease and O&D CEDAR. And our last presentation before the final Q&A panel is on statistical principles for clinical drug development by Dr. Mark Levinson, Director, Division of Biometrics 7, Office of Biostatistics, and the Office of Translational Sciences, CEDAR. Please join me to welcome Dr. Nasir.
Good afternoon, everyone. My name is Shabnam Nasir. I'm a medical team leader on the Division of Anti-Infectives here at CEDAR FDA. And today I'll be talking about safety considerations in clinical drug development. Here are our learning objectives. We want to try to understand that protection of subjects comes first when trying to design early clinical studies, and to learn some key safety considerations in the conduct of phase 1 trials, and also understand the principles of safety monitoring and reporting in clinical trials. Here are the objectives of phase 1 trials. We want to assess the safety and tolerability of the drug product, try to attempt to characterize dose-limiting adverse reactions, determine the maximum dose that's associated with an acceptable safety profile, characterize PK parameters, and explore drug metabolism and drug interactions. The subjects in phase 1 trials generally tend to be healthy volunteers that have less confounding factors because they tend to not have medical problems or be on many medications. And in some instances, patients can be enrolled, especially when the drug is known or expected to be toxic. And an example would be with cytotoxic agents. Patients, of course, will have confounding factors, and there will be more difficulty in separating disease-related manifestations from adverse reactions. Some phase 1 studies can also enroll special populations, especially if that information will be helpful for drug development, such as what happens to PK parameters in patients with renal impairment. Generally, when one is moving from the non-clinical to first-in-human studies, we have to consider what's the evidence that we have garnered so far from the non-clinical studies. For example, what's the duration and the total exposure proposed in humans, and how does that correlate with what was tested in the animals? What are the characteristics of the test drug? For example, is it a small molecule? Is it a biologic? Does it have a long half-life? What is the disease being targeted for treatment? Because that has to be taken into consideration in the benefit-risk assessment. What are the populations in which the drug will be used? Are we going to be enrolling women of childbearing potential or children in the studies? And then again, what is the route of administration? Is it going to be administered topically or systemically? So one has to consider all of these factors. Furthermore, do the non-clinical studies that have been done so far provide sufficient data to allow for the proposed clinical trials to be undertaken? What species were they studied in? Are the species appropriate and relevant to the particular drug? Were there potential target organs of toxicity identified? That's always very important. What duration was studied in the animals? Was the dose and route of exposure appropriate and similar to what is being proposed in human studies? Were the PK and PD assessments appropriate? Was there a dose response identified? And again, do we need any special studies in animals if one is planning to enroll children or pregnant women? It's important to keep in mind that only some toxicities that are seen in non-clinical studies will actually translate into adverse reactions in humans, while others do not. So you end up having both predictable and unpredictable toxicities, and these can appear in any phase of development. Sometimes you only actually see them post-marketing. It really just depends on the size of your safety database that's available pre-market. We also have to keep in mind that certain adverse reactions are subjective, and examples of this are headache or other events like hypersensitivity reactions, and they cannot really be assessed in non-clinical testing. Wanted to give you an example of a predictable toxicity from the anti-infective world, where I reside. And I'm sure you can find other examples in other therapeutic areas. So linezolid is an antibacterial drug that was approved in the late 1990s. And at that time, it was the first member of its class. It had activity against gram-positive organisms, including some resistant ones, methicillin-resistant Staphylococcus aureus, or MRSA. So certainly there was some therapeutic benefit that could be offered by this product. The non-clinical studies that had been conducted showed that there was myelosuppression, which appeared to be dose and time dependent. And there was various changes in the bone marrow, which affected all three of the cell lines. But because the product had the potential to offer therapeutic benefit, further development was pursued. When we went into phase 3 trials, there was no evidence of myelosuppression per se, but there was a high rate of thrombocytopenia seen in patients that were treated with linezolid. At the time the product was approved, the prescribing information included precautions for thrombocytopenia, and the animal pharmacology sections describe the hematopoietic effects that were noted in the animals. After the product was approved, there were cases of myelosuppression affecting all three cell lines that were reported. For example, leukopenia, anemia, pancytopenia, and thrombocytopenia were observed. So the labeling was then updated to include a warning against myelosuppression. So this, again, was an example of a predictable toxicity. Now here's an example of unpredictable toxicity. We had two products, both which were members of a beta-lactam class, which is a very well-known class of antibacterial drugs for which we have a lot of experience in humans. The structures of these beta-lactams, however, were modified to enhance the spectrum of activity. And in animals, there had been no unexpected toxicities, so both of the sponsors proceeded to conduct their phase 1 trials. The single dose actually went well, but then when they went into the multiple dose trials, subjects developed moderate to severe skin reactions, such that the product development had to be halted. So even though the non-clinical studies appear to be pretty benign and you don't see any adverse events, it doesn't mean that you won't see it when you go into humans. Next, we'll move on to the concept of a maximum recommended starting dose. There are key principles in selecting the MRSD. One is that you want to avoid toxicity at the initial clinical dose, but at the same time you want to allow reasonably rapid attainment of the trial objectives, and usually those are tolerability and PK, so that you're not so low that it will take you forever to get where your targeted dose was. Typically, we use an algorithmic approach based on what doses were given to animals and what toxicities were observed. There are some alternative doses based on animal PK and modeling, but those should be discussed with FDA prior to dosing. Some key concepts when you look at the MRSD are the no observed adverse effect level. And this is the highest dose tested in animal species that did not produce a significant increase in adverse effects compared to a control group. The second term is the HED, or the human equivalent dose, which is really what is the dose in animals that converts into a certain milligram per kilogram dose in humans. Based on that, you would be able to come up with the starting dose. You also have to consider which animal species you use to make these calculations. Ideally, you would choose the most sensitive species, and this is a species from which you can identify the lowest HED. But in some instances, you may actually want to use the most appropriate animal species because that species may be the one that has the particular target of interest. And this is especially seen with biologic drugs. Here's a schematic that shows the steps in determining the maximum recommended starting dose, and this would be usually done with the help of our colleagues at FDA from pharmacology and toxicology. So first you would determine the NOEL, which is a starting point for determining a safe starting dose for new molecular entities in healthy volunteers. Again, that you would convert that into the HED. You would select the HED from the most sensitive or appropriate species and then choose a safety factor. You would then divide the HED by that safety factor to come up with the recommended maximum recommended starting dose. And the guidance for industry references here, but it'll also be at the end for your convenience. What is the safety factor? It's the number which gives you a margin of safety for protection of human subjects when you're moving from animal studies to humans receiving the dose. The default number is 10, but there is some degree of flexibility. Sometimes we can go higher or lower. The safety factor basically provides us some reassurance that when moving from animals to humans that we're not exposing human subjects to any great risk. And one knows that there is variability when you move from animals to humans, and the safety factor allows for that variability to be accounted for. As mentioned previously in my examples, there are uncertainties when you go from animals to humans. There can be differences in the sensitivity between humans and animals to certain products. You can have difficulty in detecting certain toxicities in animals, such as headache or myalgia. There can be differences in receptor densities or affinities. You always have to be prepared for unexpected toxicities and also interspecies differences in how the product is handled. Humans and animals may not always handle the product in an identical manner. When would you increase the safety factor greater than 10? So some of the reasons are here. If it's a brand new class and we have no prior experience with the product, the toxicities in the non-clinical studies were severe or irreversible, or if they were non-monitorable. For example, if you have histopathological changes but they're not monitored because there are no clinical methods to monitor the toxicities, or there are no biomarkers, that could be a reason to increase the safety factor. If there's a very steep dose response curve, that may indicate a greater risk in humans. If the PK is not linear, or if there's variable bioavailability because you have poor bioavailability in the animals, that may actually underestimate the toxicity in when you move into humans. On the other hand, you may actually be able to decrease the safety factor to less than 10 if you're dealing with a member of a well-characterized class of drugs, or if the toxicity that's produced is easily monitored, reversible, predictable, if there was a shallow dose-response relationship, or if the NOEL was determined based on toxicity studies of longer duration, you may have a bit more reassurance in reducing the safety factor. Now let's discuss an example of a calculation of the MRST so you can get an idea. Here's a product for which the HED derived from rats was about 400 milligrams. The sponsor proposed a starting dose of 100 milligrams, so that brought the safety factor to about 4. The rationale provided was that this was a member of a well-characterized class of drugs, so using a factor of less than 10 was okay. They also noted that there was toxicity studies in 2 species that were of a longer duration than what was proposed in the clinical trials, and that these toxicities were readily monitorable and reversible. So it all sounded good. However, we did not agree with this rationale because we knew that members of the class had exhibited more toxicity than the parent class from which it was derived. And we were also concerned that the bioavailability in animals was low and that human bioavailability could be greater, leading to a risk of higher exposure in humans. So we agreed upon a starting dose of about 50 milligrams, which brought the safety factor higher to about 8. Now we'll move into some general considerations when you're doing these phase 1 trials. Some of the questions you have to ask yourself when you're getting ready to these trials are listed here. Are the clinical trial protocols designed appropriately to ensure safety and meet the stated objectives? Your job is to try to protect the safety of the subjects in the trial at all times. Do you have enough information about the quality of the investigational products? And at FDA or OPQ or Office of Pharmaceutical Quality and Chemistry Manufacturing Controls, they look at this information. The quality, has the formulation been well characterized, and do we know what the impurities are? Are the route and rate of administration appropriate? Can the product be given as a bolus dose, or does it have to be given as a slow infusion because the toxicities are such? What is the mechanism or mode of action? Is it a novel mode of action, or do we know anything about similar products that act via a similar mechanism? And then lastly, are you expecting an off-target effect? And this is a very important step if you're, for example, if you're dealing with a biologic drug. The mode of action may involve a target that's connected to multiple signaling pathways or affects a biologic cascade or cytokine release. So the mechanism of action in that case will be even more important in terms of safety considerations. So how do we determine an appropriate dose? Ideally, one subject should receive a single dose, and then that's followed by sequential administration within each cohort. You want to ensure that there's adequate periods of observation between dosing so that one has the time to observe and interpret adverse reactions in the cohorts at the lower dose. And how long you observe will depend on the properties of the drugs. For example, the PK properties, as well as how long does it stay in the system, how long is its half-life. We also learn some very good lessons from previous trials conducted with similar products. So prior knowledge can be helpful. If the protocol for these phase 1 trials allows for dose escalation, you have to ask yourself whether the dose escalation is appropriate. Are the dose increments appropriate? Is it okay to double it up or go up by 4 times? You do have to be cautious and use a slower rate of dose escalation if you have a small therapeutic window, or if your animal models were not very good, or if you have any particular concerns about toxicity. And there is a fair bit of flexibility here. It all depends on your product, on your target, and the toxicities that were seen in the animals. You have to ask yourself, is the amount of information before each dose escalation appropriate, and are the number of subjects at each dose appropriate? It's important that the protocols have an appropriate monitoring scheme to look at clinical signs and symptoms of adverse events likely to be associated with the drug. The duration of the clinical observation should also be adequate with respect to the stated objectives and the endpoints, the anticipated response to the product, and the health-related conditions that are being studied. Sometimes we do need to monitor for longer periods of time, but again, it all depends on the characteristics of the drug. You may need a hospital setting for some drugs that are being studied following initial dosing. If you're worried about any sort of delayed onset reactions, undetected toxicities, you want to make sure that the follow-up is long enough as well. As far as the frequency of monitoring, there may be a need to observe more frequently soon after initial dosing in the first week or two, and certainly you would need follow-up visits for subjects who develop adverse events or lab abnormalities. In addition to monitoring clinically for adverse reactions, you would also need to monitor for lab tests, and which particular lab tests you order and collect the data on really depends on your drug, again, the type of target organs of toxicity. So you may want to ask yourself, do the laboratory assessments, will they need to include routine assessment of all organ systems? Do we know enough about the drug that you may only need to assess specific organ systems, like liver or kidney? And are they sufficiently detailed so that you can make an assessment of whether or not the drug is causing toxicity of those specific organs? And lastly, are there stopping rules based on laboratory abnormalities if they reach a certain threshold? Now let's talk about safety stopping rules, because these are very important. So again, it's important that the protocol has clear stopping rules for administering the drug, the enrollment, the dose escalation, and termination of the trial. Sometimes we've actually had to put INDs on hold because there were no stopping rules. So again, these are changes to the protocol that are to be implemented when toxicity is observed. To be able to have stopping rules, you have to identify what might be acceptable toxicities and what are not acceptable toxicities. Since it's unlikely that you'll have a drug without any toxicities, it's important to think about these. If acceptable toxicities occur, these will not result in changes in how the trial is continued in terms of subject enrollment and dosing. If you have other toxicities that are not acceptable, you have to have a procedure in place for what you would do with the study. Challenge question number 1, to take a little break. Which of the following is used to calculate the MRSD? Hopefully you recall that the key step in this process is to determine the NOEL, or the no observed adverse effects level, which is A. So hope everyone got that one. Now we're gonna move a little bit to the actual evolution of safety as you go down further into your clinical development stage. Evaluation of safety is an evolving process. It depends on the stage of the product under development. For products that are approved or marketed, the safety information is reflected in the product labeling. This is also called the prescribing information, or PI. And up-to-date safety information for investigational products are listed in the IB, or the investigator brochure. And the investigator brochure will talk about what kind of information is available there. So sources of safety information can be either found in the IB or the prescribing information. And the types of sources can include non-clinical data, in vitro or animal data, as well as information about the structure of the product and the manufacturing process. Clinical pharmacology data can include information about PK/PD. Safety information can come from those early clinical studies that we discussed from healthy volunteers and patients. But of course, probably the most pertinent information will be the data from the actual clinical trial for the same indication, as such data is generally controlled for the patient population characteristics and underlying pathologic process. You can also have clinical trial data for different indications, post-marketing experience, the published medical literature, and last but not least, the safety profiles of drugs in the same class. So all of these can be contained in the IB and or the PI. And the IB would actually also provide a description of possible risks and adverse drug reactions to be anticipated based on prior experiences with the investigational product and with related products. Now, why is safety monitoring required in all trials? The goal is to identify, evaluate, minimize, and appropriately manage risk. And so this will lead well into our discussion of adverse events ascertainment and monitoring, which is sort of the second half of my talk. What do we consider an adverse event? So according to the CFR that I have listed here, it's any untoward medical occurrence associated with the use of a drug in humans, whether or not it is considered to be drug-related. This could be any unfavorable sign, symptom, disease, abnormal vital sign, abnormal laboratory finding, imaging, or any worsening of the above or constellation of the above. Adverse event can also be referred to as an adverse experience. Some examples of ways that adverse events can be ascertained include spontaneously reported or observed symptoms or signs. They can be symptoms or signs reported as a result of a probe, such as a checklist or a questionnaire. These can also include various types of testing of laboratory results, looking at CBCs, liver tests, etc. You can have specific safety assessments conducted depending on what you think is important in the clinical trial, such as visual, hearing, neurological exams, and again, ascertainment of vital signs. And adverse events can be characterized according to their severity. There's a number of severity grading scales or tables that are available to provide guidance to investigators for safety monitoring in clinical trials. And these are optional tools, but they can be helpful. They are specific to the study population being looked at. So, the grading scale will be different depending on whether there's patients enrolled versus healthy volunteers, what is the phase of product development, and the product type to be evaluated. For example, in healthy volunteers, clinical abnormalities may be represented on a scale from mild, moderate, severe, or potentially life-threatening, whereas, for example, in oncology patients, AEs may be graded by a scale of 1 through 5, whereas 1 is mild symptoms versus up to, grade 5, which is death related to the AE. So it really depends on your study population as well as some of the other factors I've noted. And some of the commonly used examples are listed here. It's also important to keep in mind that the term severe is not the same as serious in the classification of AEs, and so we will be talking about SAE later in the talk. What happens when the AE report gets to the sponsor? There's a process of converting from the investigator's verbatim terms to the standardized preferred terms using a specialized dictionary called MEDRA. MEDRA is the medical dictionary for regulatory activities, and it can be used to sort AEs and group like events. Preferred terms are usually what are used to calculate the incidence of AEs. And wanted to show you a structural hierarchy of the MEDRA system. The highest level of terminology, which is the least specific, is the system organ class, or SOC, and this goes down to the lowest level term. The PT is the one that, again, is most important, which because it represents a single specific medical concept. Clinical investigators actually play a pretty crucial role in the safety reporting because they have to try to use the most scientific and consistent terminology when reporting adverse events. And there may be significant coding problems that may lead to missing safety signals. So one example is splitting, where if you split similar terms such as hypertension or high blood pressure, it can actually result in a lower incidence or may minimize or mask a safety signal. Lumping, on the other hand, lumping dissimilar terms to the same PT may also obscure a safety signal under the lumped term. So an example of this is facial edema, which can be a very different type of pathophysiology from leg edema, for example. And a coding issue could be that one has lumped all edema to the PT edema. In the third category, there may be a lack of adequate term or a definition to represent a syndrome of interest, such as serotonin syndrome or metabolic syndrome or hypersensitivity. And in this case, you may actually need a prospective case definition if the syndrome is not well characterized by a single term, especially if you're expecting one of these to occur in your study. So here is our next regulatory definition, which is serious, not to be confused with severe. Serious adverse event, and the CFR is listed here for more information. These are ones, AEs that result in the opinion of the investigator or sponsor to result in death, life-threatening event, in hospitalization or prolongation of hospitalization, a disability or birth defect, congenital anomaly, or any other important medical events, which are usually those that jeopardize the patient or require intervention to prevent one of the outcomes listed above. And an example may be an allergic bronchospasm requiring intensive treatment in an ER or at home. Here's some examples of uncommon SAEs. These should really raise a red flag that there may be a possibility that the AE was caused by the study drug, especially in the study population of generally healthy individuals. You don't want to see one of these occurring. While this isn't a comprehensive list, it is a good list to kind of have with you. And some of them can include anaphylaxis, liver failure, seizures, torsade de pointes, and others listed here. So serious adverse events, like any other adverse events, need to be assessed for drug-related causality. Some of the questions that you can ask yourself to determine this would be, is it common in the population under study? Was it treatment emergent, or did it occur on treatment or shortly thereafter? Did it respond to de-challenge or a drug discontinuation? Did it occur on re-challenge or reintroduction of the study drug? Were there any concomitant medications on board that may have contributed to it? And then were there labs or tests done to characterize the adverse event further? Was there any obvious alternative cause? And then lastly, is the SAE a study endpoint? So for example, was death in your study also a study endpoint? And you want to keep in mind that you want to try to provide enough detail in relevant clinical information in the case report form to allow for a good quality narrative to be generated. So now let's talk about AE reporting requirements, what the clinical investigator would report to the sponsor, and the CFR is listed. And this is also discussed in a draft guidance for which the reference will be provided at the end. An investigator must report immediately all SAEs whether or not the event is considered drug-related. In addition, the investigator must also include an assessment of whether there is a reasonable possibility that the drug caused the event. For SAEs that are also study endpoints, they can be reported in accordance with the protocol. However, one exception to that is that if the study endpoint is an SAE and there's evidence suggesting a causal relationship between the drug and the event, the investigator then must immediately report the event to the sponsor. Okay, so let's talk about our first hypothetical case. You're the clinical investigator for a trial evaluating whether antihypertensive drug A is associated with a reduced risk of death, MI, or stroke. A 75-year-old white male patient has just died in your trial. And we are told a little bit of information about the cause of death, which appears to be anaphylaxis. So the question is, do you have to immediately report this case to the sponsor? So let's think just for a minute. So we're told that death is a study endpoint, and usually SAEs that are study endpoints can just be reported per protocol. However, here we're told that the cause of death was due to anaphylaxis. So anaphylaxis is a pretty rare event, and as you noted on the previous slides, it's one to kind of raise a red flag that there may be a causal relationship with the drug. So now, as you have evidence to suggest a causal relationship between drug A and the event, the investigator must immediately report the event to the sponsor. So this was this is a little tricky, but I hope that this explanation comes through. Now moving back to regulatory definitions, this is the one that's important for the expedited reporting of adverse events to the FDA. And the concept of unexpected adverse event is presented here. There's a lot here to unpack, so the CFR may be good to review for this one. An adverse event is deemed unexpected if the following conditions are met. If it's not listed in the investigator brochure or IB, or if the IB is not available or required, if it's not listed at the specificity or the severity observed, for example, if the IB lists increased creatinine but you have a case of acute renal failure, which would be unexpected by virtue of greater severity, or if you have a cerebral thromboembolism, that would be unexpected by virtue of greater specificity if the IB lists only a CVA. If it's not consistent with risk information described in the general investigation plan or elsewhere in the current application. And then lastly, also I did mention if it occurs at a higher rate than listed in the IB, that would be another reason for unexpected. And lastly, if it's mentioned in the IB as anticipated due to the pharmacological properties of the drug, or if it occurred with other drugs in the class, but not with the particular drug under investigation, it would be considered unexpected. So an example would be angioedema. It's anticipated to occur in some patients exposed to drugs in the ACE inhibitor class. And angioedema would be described in the IB as a class effect, but if you have a case of angioedema observed with the drug under investigation, that would be considered unexpected for reporting purposes until it's included in the IB as occurring with the actual drug under investigation. So now let's look at another hypothetical case. Here you're the investigator for a clinical trial evaluating a new quinolone antibacterial drug B for the treatment of pneumonia. The investigator brochure lists a number of SAEs associated with the quinolone drugs, including essential tremor. Again, this is hypothetical. Is the seizure in this trial for drug B considered an unexpected AE? So yes, seizure in this hypothetical case would be considered unexpected because the IB does not list seizure. Or you can also say that the AE seizure is not really at the severity of what's listed in the IB. The seizure is at a greater severity than what's listed in the IB, which is just essential tremor. All right, so I hope that was an illustrative case. Let's keep moving on to our next and final regulatory definition, which is suspected adverse reaction, or SAR. And this is an AE for which there's a reasonable possibility that the drug caused the AE. Again, reasonable possibility means that there's evidence to suggest a causal relationship between the drug and the AE for purposes of IND safety reporting. Some examples can include the following: a single occurrence of an uncommon event that's known to be strongly associated with drug exposure, for example, angioedema, hepatic injury, a Stevens-Johnson syndrome case. Or more than or equal to one occurrence of an event not commonly associated with drug exposure, but otherwise uncommon in the exposed population. For example, neutropenia in a healthy subject would fit sort of into this example. Or an aggregate analysis of specific events observed in a clinical trial, indicating that those events occur more frequently in the drug treatment arm than in a concurrent or historical control group. So for example, acute MI in a long duration trial with elderly cancer patients would be not so unexpected. So you would want to have an aggregate analysis of these specific events to to say that this was a suspected adverse reaction. Okay. Determination of an AE as a suspected adverse reaction can be difficult, but it's critical to avoid submission of uninformative IND safety reports. The sponsor is the one that evaluates all available information and decides whether there's a reasonable possibility that the drug caused the AE. So again, this is the sponsor's primary responsibility. This slide illustrates the universe of suspected adverse reactions, which are no longer just adverse events, which are in the outermost sphere here. So these are adverse events are again, they have undetermined causality. And they're not quite, suspected adverse reactions are not quite established, definitively established adverse reactions that are noted in the dark red sphere in the center. So these suspected adverse reactions occur in the middle here. And again, these are AEs with a reasonable possibility of drug-related causality. That is, there's evidence to suggest a causal relationship between the drug and the AE. Okay, so FDA continues to require that 3 criteria generally be met before a sponsor submits an IND safety report. And these criteria again are that its event must be serious, unexpected, and there must be evidence to suggest a causal relationship between the drug and the event. So all three criteria have to be met, and the standard is sometimes called a SUSAR, serious, unexpected, suspected adverse reaction. And then again, the sponsor is the one that must assess whether or not there's a reasonable possibility that the drug caused the event. The sponsor should only submit these type of IND safety reports when all three criteria are met. In addition, the sponsor is expected to submit an annual report to the IND, and this will include a summary of the most frequent adverse events that are not reported individually, in addition to summarizing the SAEs. So let's talk about time frames for reporting. Sponsors have two time frames when it comes to IND safety reports. Basically, those that are required to be submitted to the FDA in 15 days and those that are required to be submitted within 7 days. As you can imagine, the most serious ones are those that are reported within 7 days. There's 5 categories of adverse events that trigger the sponsor's responsibility to submit these. So one, again, is the SUSAR that we just mentioned. Serious, unexpected, suspected adverse reactions. Findings from other studies suggesting a significant risk in humans. Findings from animals or in vitro testing suggesting a significant risk in humans. An increased rate of occurrence of serious suspected adverse reactions. And serious adverse events from bioavailability or bioequivalence studies not under an IND. And so for all of these, this IND safety report would be required within 15 days unless the adverse reaction was fatal or life-threatening, in which case the reporting would be required within 7 days of submission. And there may be some details here that you want to look at, and the guidance on this reporting will be, again, provided at the end. Now, this is our final example, hypothetical case number 3. In a trial of a marketed HIV drug, C, an 8-month-old infant enrolled at 1 month of age was noted at month 4 to have moderate hearing loss in a clinic progress note. Ototoxicity is not mentioned in the labeling of this drug class. The investigator's causality assessment notes that hearing loss may be related to the study drug. So the question is, does the sponsor have to report this to FDA? And if so, can it be submitted as a 7 or 15-day report? So in answering this question, we have to think back to our definition about a serious adverse event. In this case, hearing loss is a disability and thus it's an SAE. As hearing loss is not mentioned in the labeling, it is an unexpected event. The sponsor determines that there's a reasonable possibility of relatedness to study drug. We're not told, but we also have information. The sponsor has information that there was an increased rate of occurrence of hearing loss on the study drug as compared to the comparator. So with all that information in mind, the sponsor determines that there is a reasonable possibility of relatedness to the study drug, and it qualifies as a SUSAR, and thus it should be reported to FDA. As it's not fatal or life-threatening, it can be actually reported as a 15-day report. So in summary, I've provided a high-level overview of safety considerations in phase 1 trials, important questions to ask yourself before you decide to dose humans as you go from translating information from non-clinical studies to either healthy volunteers or patients. We talked about the relevance of toxicities seen in the non-clinical studies and how they translate to adverse events in humans. It's important to keep in mind that they're both predictable and unpredictable adverse reactions. We discussed how to go about determining a safe starting dose in humans and the concept of the safety factor. We also discussed the importance of safety monitoring and stopping rules. And we moved on to the ascertainment of safety in larger clinical trials and important regulatory definitions of adverse events. Investigators play a critical, integral part in assuring quality safety assessments by then reporting also to the sponsor. And we also learned how the sponsor evaluates all available safety information and reports to FDA and all participating investigators. So my last slide is a challenge question. An investigator would not have to report a case of hepatic failure if the investigator brochure listed elevated hepatic enzymes or hepatitis. And I think you will all get this answer. This is false. Again, because the case of hepatic failure is at a higher level of severity than what's listed in the IB. And with that, I thank you for your attention and have a great rest of your day. Thank you.
Good afternoon, everyone. My name is Mark Levinson. I'm a statistician in Cedar FDA. I'm happy to be speaking today at this clinical investigator training course. And I'll be speaking on statistical principles for clinical development. Okay, here are my learning objectives. The first objective is quite broad, to understand statistical principles for the design, conduct, and analysis of clinical studies. The second two objectives are more specific, to understand the concepts of bias and variability, and to understand issues around adherence, missing data, and multiplicity. Now, naturally, in a half hour, people aren't going to go from no understanding to being experts on all this, but I hope this talk gives you a good basis of the key concepts, and I do have references throughout for further learning. Okay, before I go too far, I'd like to define these terms on the stages of a study. And before I do that, I'm introducing my first references here, ICH E8 and ICH E9. So ICH is an organization made up of regulators and industry groups from around the world, and among other things, they produce guidelines on topics related to clinical trials and drug development. ICH E8 is on general principles for clinical studies, and ICH E9 is on statistical principles for clinical studies. And I provide more details on these references on a slide at the end. Okay, so design. Design is the conception, planning, and specification of the study. Conduct is the actual running of the study. Analysis is the analysis of the study, or sort of the number crunching. And this is the part where people feel the statisticians play their role, but the point of my talk and what I'll argue throughout is statistics has a role in all these stages. And finally, there's reporting, which I won't really have anything to say about today. So here's kind of the key takeaway maybe from the whole talk that in some ways is the principle that everything else is based on here, is that design and conduct are more important than analysis. In other words, analysis cannot make up for poor design and conduct. Thinking about it in sort of the positive direction, if you focus on good design and conduct, the analysis will be straightforward. Okay, there's a lot on this slide. Let me back up a bit. In order to market a drug in the US, you have to demonstrate that the drug is safe and effective. You also have to demonstrate other things as well. To demonstrate a drug is effective, you need evidence from adequate and well-controlled studies. And what defines an adequate and well-controlled study is specified in federal regulations. And on the bottom is the reference to those regulations. And they're actually surprisingly readable if you ever want to look those up, if you Google that reference there. Now, even if your goal is not to market a drug, but do some scientific study outside of regulatory purposes, these are good scientific principles regardless. So let me go through them somewhat quickly because some of them I'm going to go into greater depth later. So clear objectives, summary of methods, and results. So naturally, when you begin a study or plan a study, it should have clear objectives, and the approaches you use should be clearly specified. The design permits a valid comparison with the control. That one is key, and I will spend a few slides on that, so let me go on. Adequate selection of patients. So that means the patients in your study should have the condition you're interested in. That seems kind of pretty straightforward, but there are cases where there might be challenges there. Assigning patients to treatment and control groups to minimize bias. This is where randomization comes in, and I'll return to this one as well. Adequate measures to minimize bias on subjects, observers, and analysts. This is mainly about blinding, and again, I'll return to this one as well. Well-defined and reliable assessment of subjects' responses. So this is like getting good outcomes and endpoints from your studies. And it's a very important part of the design and conduct of a study, and sometimes overlooked. I believe there are other talks in this training on this topic, and I really won't have much to say about this, but I will highlight that it's an important part of the design of a study. And finally, adequate analyses to assess drug results, and that's where statistics is sort of front and center. Okay, let's drill down on the purpose of a control group. And here I reference ICH E10, and that's on control groups. And it's an excellent document, good reading. These are accessible documents, these ICH documents. So what is the purpose of a control group? Almost verbatim from ICH E10, it allows for the discrimination of patient outcomes caused by the test treatment from other factors. So the alternative of having a control group perhaps is like a single arm study where you just have patients on the test treatment. And in certain circumstances, that's sufficient, but that's probably the exception. In almost all cases, a control group is needed. So without a control group and you just had this single arm and you observe that patients got better, could you attribute that to the test treatment, or are there other factors? So, for example, people get better or worse on their own, the natural progression of the disease. You don't know if it's a test treatment or just a natural progression of the disease that's causing the improvement. Observer or patient expectations. With a single arm, obviously people are going to know what therapy they're on. And it might just be their expectations. I'm on this brand new experimental drug. I know I'm getting better. So it might be their expectations. And if there's any subjectivity to the outcome, and there often is, there might be a bias to better results because of patients' expectations. And concomitant treatment. Now, the test treatment is not the only therapy or medical intervention that patients are likely getting. There's other therapy going on, and it's possible this other therapy, this background therapy, is actually, causing the effect that you're observing. So that kind of summarizes the big purposes of the control group. So just argued that the need for control groups, but it's important that the control group is as similar as possible to the test drug group, except for the test drug itself. And that similarity should be both at baseline and at post-baseline. And to get the similarity at baseline, that's where we use randomization. To get similarity post-baseline, that's where concepts such as blinding, focusing on compliance, focusing on complete data and follow-up, and focusing on good outcome come into play. And I do reference ICH E8 here for a little bit more on outcome assessment. So let me go further into randomization. So I'm going to contrast a randomized versus an observational study. And this is mainly to bring out the sort of benefits of randomization. So in a randomized study, a patient comes along, and through some random process, here just for illustrative purposes, shown by a flip of a coin, a patient is assigned either to drug A or to drug B. This is in contrast to an observational study where the study doesn't intervene on what drug the patient gets. So in an observational study, the healthcare provider, in consultation with the patient, chooses the best drug for the patient. And that decision may be based on lab tests, it may be based on demographics and medical and family history, other drugs the patient may be taking, and also may be based on maybe practical things like insurance and convenience. But the patient factors concerning the patient go into the decision of what drug they get. This brings us to the concept of confounding. So without randomization, there may be systematic differences or biases when comparing people getting drug A versus people getting drug B, and this is known as confounding. So for example, suppose drug A is given to older, sicker people. Perhaps drug A has less side effects and is better tolerated. So for older, sicker people, it might be the better choice. So if even if there were, even if there were no differences between the effects of drug A and drug B, the comparison may show that drug A has worse outcomes. So I hope this is kind of a quick motivation of the reasons and benefits of randomization. Okay, just a quick aside here. I would hazard to say many of the studies you hear in the news are observational studies. Take them with a grain of salt, to say the least. As a rule of thumb, anything you hear about coffee, red wine, or chocolate, it's probably not based on strong evidence. Now, I don't want to kind of badmouth observational studies. They do have their place. They can be done well, and there are certain settings they are appropriate, and FDA does make use of them in important regulatory decisions. Okay, let's move on to blinding. Blinding is discussed in ICH E10, E8, and E9. So the double-blind situation. Here, patients, investigators, and study staff do not know treatment group membership. So this minimizes differences in both the patient management and the assessments, both on the efficacy and safety side. So I'm not claiming that if things are unblinded, everyone is consciously biased and just trying to make the drug look as best as possible. But biases are often unconscious. That's why blinding is important. But I will say that even with blinding, patients or investigators may guess what drug they're on. There may be certain obvious side effects of a drug that makes it clear to the patient or the investigator that a patient's on a particular drug. So just because you didn't tell the investigator or the patient what drug there are, you can't automatically assume that they're completely ignorant of this. So this also should sort of be accounted for in the design of the study. For example, in the assessments, the patient outcome assessments, may be better done centrally or somebody unrelated to the management of the patient, but just something to be aware of. Okay, I've already used the term bias a number of times. I don't believe I've used the word variability, but let me kind of make these terms more specific here. I think this is useful. So here we have these targets, and the center of the target represents the true value, the value we're kind of interested in, but don't know, and the purpose we're doing the study to find out about. And the dots represent our data points. So starting in the upper left, we have low variability and low bias. So we see that the data points are generally around the center of the target, so that's low bias, and they don't have much spread, so that's low variability. Moving down, lower left, we have low variability but high bias. So here the data points are close together in the low variability, but they're far from the true value, so that's high bias. Moving to the upper right, We have high variability, that the data points are spread apart, but low bias because they're generally centered around the true value. And finally, on the lower right, we have high variability and high bias. And this is, of course, the worst case. In practice, we don't see the targets. And what that means is we don't know if we're in a bias situation, a low bias situation, or a high bias situation. We do see the spread of the data, so we know the variability situation, and we can measure that. But we don't know if we're unbiased or in a situation with high bias. And that's why it's important in the conduct, or the design and conduct of the study to do everything possible to minimize important biases. And that's why it's important to do randomization, to do good conduct with blinding and good outcome ascertainment, because bias sort of is an unknown unknown, and we want to avoid it when possible. I've spoken mainly about bias so far, but I do want to just say something about variability. There are several ways or multiple ways of reducing variability. One of probably the key ways of reducing variability is to have more sample size. So more sample size means more people in the study. So I think everyone has a understanding of this. So what is a better estimate of the average age of this session's attendees? You can pick a random sample of 5 attendees and calculate their average age, or you can pick a sample of 20 attendees and calculate their average age. And naturally, B, with more attendees, is going to give you a less variable estimate, irrespective of any biases. Now, in practice, it's not always possible to have larger and larger sample sizes, so feasibility needs to be accounted for too. So in summary about variability and bias, statistics can help quantify variability in the design and analysis stage. So in the design stage, it's a lot about what the optimal designs are and sample sizes are. And in the analysis stage, statistical methods calculate things like confidence intervals and p-values that help quantify variability. Design and conduct generally reduce bias, and we've already seen that randomization, blinding, and good outcome assessment are generally how this is done. And finally, statistical principles help reduce bias and variability at each stage: design, conduct, and analysis. Now I will speak about adherence and missing data issues. Adherence and missing data are one source of bias in a clinical study. Before I continue, let me note that the material and example I use here are adapted from work of Dr. Susan Ellenberg, a renowned statistician who I believe taught this course previously. So what may happen to a patient in terms of adherence during a clinical trial? Well, the patient may stop treatment. This is not necessarily a bad thing. It may be in the best interest for a patient to stop treatment because of side effects or other medical conditions. In this, the patient may stop treatment, but they may or may not be continued to be followed up in the trial. Patients may leave the study. In this case, what I mean is there's no more follow-up. The patient has left the study and is no longer participating and cannot be followed up any further. Or a patient may miss an individual study visit, and that study visit might have consisted of a treatment or an assessment. So what are some causes of adherence and missing data? Well, it might just be random, and by random I mean it's not related to any sort of patient characteristics or the patient outcome. I mean, using an extreme case, maybe there was an earthquake in the region and the clinical site has to close down. Maybe it's related to the frailty of the patient. Perhaps sicker patients are unable or unwilling to continue to be part of the study. Or maybe it's related to lack of efficacy. A patient who perceives that they're not getting benefit from a therapy may choose to stop therapy or may choose to stop being part of the study altogether. And likewise, a patient who's experiencing side effects may decide not to continue treatment or more extreme may decide not to be part of the study. So some important points of how this all introduces bias. Patients with complete data may be different from those with incomplete or missing data. We saw in the last slide, well, if it's random, maybe the patients with complete data are similar to the patients with incomplete data. But if it's related to efficacy or frailty or adverse events, it's likely the patients who complete treatment or complete the study are different from those who do not complete the study. What's a more kind of subtle point, which we probably don't have time to go into now, is treated group patients with complete data may be different from control group patients with complete data. There may be different reasons why treated patients drop out versus why control patients drop out. So in general, analyzing patients with complete data will likely produce biased results. Now I'm going to move on to a famous example that maybe demonstrates some of this and with some real data. This is based on a trial that compared clofibrate versus placebo for lipid lowering. And the outcome was mortality here. So on the bottom, you can see 18.2% of the clofibrate had mortality versus 19.4% of the placebo. This was a small, non-statistically significant difference. However, a significant proportion of the patients on clofibrate did not have perfect adherence, which is not surprising. So about a third of the clofibrate patients had less than 80% adherence to the treatment. If you look at only those patients who had greater than 80% adherence, the mortality drops to 15%. So you compare that 15% to the 19 or so percent of placebo, you start to see that maybe the drug does have an effect. However, now I reveal the whole table. It looks like the placebo patients who had greater adherence have lower mortality as well. So the patients who have greater adherence are associated with lower mortality. And I'm sure we can think of many reasons why that might be the case. But for patients, whether they're in the clofibrate or the placebo arm, the mortality is 15%. So again, it's pretty clear this drug does not have an effect on mortality, at least as shown by this trial. But importantly, we see that patients' adherence is very much related to their outcome, even on placebo. So it's not the drug necessarily that's causing the difference in mortality. It's the differences in patients. What are adherence and missing data strategies? How do we address adherence and missing data in clinical trials? Two ICH documents are relevant here. ICH E9 on statistical principles that I already mentioned discusses missing data. And ICH E9 R1, an addendum to the original I9, which came out a few years ago, further discusses it. The first principle is we want to avoid missing data. So whenever possible, we want to get follow-up data regardless of treatment adherence. So if a patient stops treatment, we still want to follow them up. There are exceptions to this, but this is sort of the general principle. It may be challenging if a patient stops treatment, they may no longer be interested in being part of the study or providing follow-up data. But whenever possible, we want to avoid missing data. Our general strategy is to use the treatment policy as is referred to in E9R1 or intent to treat, ITT, as is referred previously in E9. In this strategy, patients are analyzed according to their random assignment regardless of adherence and missing data. So we're not just analyzing patients who complete treatment. We're not just analyzing patients who don't have missing data, because patients with missing data may be different from patients without missing data. We want to analyze all patients according to the original random assignment. The challenge, of course, is what do you do with patients with missing data? Well, there are a number of approaches there that generally fall under the idea of imputation. You might be able to impute the missing data based on their previous observations and characteristics of the patient. There's assumptions that go into this, but there are statistical methods that make the assumptions clear and also sort of incorporate the additional uncertainty of those assumptions. So again, this is not something we can go into depth today, but there are statistical methods to handle missing data. Besides the treatment policy approach, there are other approaches depending on the situation, and you can explore E9R1 to hear more about those. Now I'm going to move on to the last topic of this presentation, multiplicity, also known as multiple bites from the apple. And I'm going to use a toy example to introduce this. So we have a study that consists of flipping a coin 4 times. Here, heads is a good outcome, and tails is a bad outcome. If we get 4 heads, we conclude the drug has an effect. If we get 1, 2, or 3 heads, we conclude the drug has no effect. So we do the study, and the first study has 3 heads, so we conclude the drug has no effect. But we're not satisfied with the result, so we do it again. And we repeat the study, and here we only get 1 head, so it's still not good. We do it again. Here we get 2 heads, and it's still not good. It took us 12 times, but eventually we get 4 heads, and we publish the results and say our drug is effective. You know, I think you can see sort of the problems with this approach. Multiplicity can come up in many ways. Now, the toy example I gave on the previous slide is about doing multiple studies. So if you do enough studies, you'll see a result. But importantly, multiplicity can arise in a single study by looking at multiple subgroups or multiple endpoints. So subgroups might be the effect on males, the effect on females, the effect of people over 65. Multiple endpoints may be the effect of blood pressure, the effect on life expectancy, the effect on happiness. So by looking at multiple things within a single trial, you get the same sort of problem I tried to demonstrate on the last slide. So what are the solutions for multiplicity? Well, the primary solution is pre-specification. You tell the world ahead of time what you will primarily look at. And this is done through protocols and statistical analysis plans. The protocol should be in place before you start the study, and the statistical analysis plan should be in place before the data are unblinded and should follow what's already outlined in the protocol. There are statistical methods that do handle or can handle multiple outcomes or subgroups within a trial, which I, of course, won't have time to speak about today. But these need to be pre-specified too. This brings me to the summary. So I hope I conveyed these principles throughout the presentation, but careful design and conduct are needed for reliable results. Randomization addresses baseline biases. Blinding, good adherence, good follow-up, and good assessment address post-baseline biases. And pre-specification is important to address multiplicity. This brings me to the challenge questions. Challenge question 1 is, which of the following does not reduce bias? A larger sample size, randomization, blinding the knowledge of the drug from the participants and investigators, pre-specification in the protocol and statistical analysis plan. Give you just a few seconds to think about this. And the answer is a larger sample size. That will reduce variability, but not address biases. That's an important thing to consider. Just making the study larger will not make biases go away. Challenge question 2, which of the following addresses multiplicity? We have the same choices, a larger sample size, randomization, blinding the knowledge of the drug from the study participants and investigators, pre-specification in the protocol, and statistical analysis plan. And this one I think is more straightforward. I just covered it. And the answer is pre-specification in the protocol and statistical analysis plan. Here are the full titles of the ICH documents that I referenced throughout the presentation. And finally, let me leave you with this. Good scientific practices, design, conduct, and analysis will promote valid and reliable results. Thank you for your attention, and I will be available for questions at the end of this session.
Shabnam and Mark, thank you so much for those presentations. You covered a massive amount of information very clearly, so hopefully the listeners will find that helpful, as well as the resources that you shared. Moving on to the question and answer period. Shabnam, if you're there, we'll go ahead and start with you. What information goes into the investigator's brochure?
The audience, I think you could probably hear me well.
Yes, go.
Okay, thanks. So yeah, we did talk about this. So what goes in the IB? And the IB, the investigator brochure, is important component of an IND submission. So it would include a brief description of the drug substance and the formulation, including the structural formulation. You want to have a good summary of pharmacological and toxicological effects of the drugs in animals, and if there's any information in humans, you want to have a summary of the PK and the biological disposition of the drug in animals, and if known in humans. And then importantly, you do also want to have safety and effectiveness information in humans obtained from prior clinical studies. And then also importantly is having a description of possible risks and side effects that you anticipate based on the prior experience with the drug under investigation, or with drugs that are in the same pharmacological class, and also precautions or any kind of special monitoring that you want to have done as part of the investigational use of the drug.
Thank you so much, and I think that's actually a good introduction to our first session tomorrow. For those of you who are interested in more detail on this topic, we'll be covering chemistry, manufacturing, and controls, pharm tox, as well as clinical pharmacology in more detail. So some additional insight into what's included in an investigator's brochure. One more question for you, Shabnam, for now. What information should the investigator collect about the serious adverse event to report to the sponsor?
Thanks for the question from the audience and from Kim. So the investigator should collect certain information about the serious adverse event to report to sponsor, and obviously you'd want to include the specified subject, the suspected drug, and the reporting source. Sometimes it's not the clinical investigators themselves. You want to have a clinical description of the event, including an assessment of whether there's a reasonable possibility that the drug caused the event. Let me just make sure that you guys can hear me okay. Okay, sorry about that. So yes, you want to have an assessment of whether there was a reasonable possibility that the drug caused the event. So that's a very critical element that is very helpful for the sponsor in their assessment as well, to determine if it's a serious event.
Terrific, thanks. Mark, there are some questions for you here as well. So the first, I'll take a question from the audience here. Blinding, especially double blind, is a staple of the randomized controlled trial to reduce bias. Is there any place for blinding in collecting real-world data?
Well, thank you for the question. I think it's a very good question. I have to apologize. I did not attend Dr. John Concato's talk this morning on real-world evidence, but I can guess that he made a point that FDA considers RWE, real-world evidence, to include evidence that comes both from observational studies as well as clinical trials. In purely observational setting, it seems unlikely that you're going to get blinding of the outcome measure. That's not how medical practice is typically conducted. But in a trial setting, in an actual interventional setting, you can conceive that there might be some additional assessments outside the use of real-world data that's already collected that could be blinded. And that's probably an effective strategy. If a blinded outcome is important, then it's probably maybe worthwhile in sort of a pragmatic trial setting to do an additional assessment with a blinded outcome assessment. So I hope that answers the question. Thank you.
Yes, thank you. Another intention to treat question. Well, I got one earlier today. There's clearly interest in this area. What is the rationalization for an intention to treat analysis to include patients who may have been switched from one treatment arm to another, such as a patient randomized to an endovascular procedure but then switched to the medical management arm? It seems that the switch would confound the trial results.
Yeah, this is a very good question, and I think a difficult question to answer, but I'm going to try. In practice, we don't know if at baseline, when someone is given a new therapy, we don't know if they're going to switch or not switch. Likely that switch itself will be based on the effect, the safety and efficacy of the test drug itself. So it's kind of hard to separate out the switching from the effect of the drug. Now, there are procedures that are proposed to do this, and this is getting more on the research side. There's what's called per protocol analyses, and there's a good paper written on it by Miguel Hernan that tries to use the observed data, both baseline data and post-baseline data of the patient, to adjust for the switching and try to sort of get at maybe the true effect of the drug had someone been completely compliant. But it's very challenging to do. I think if it could be done effectively, there would be a lot of interest in it. But in practice, you have to know why someone switched. You have to have a clear understanding of why someone switched, as well as sort of measure the variables that indicate their propensity for switching. So it's very, I think, difficult to do in practice. So ITT is also referred to as treatment policy. It's a very simple approach. Like if you give someone a particular treatment and the actual course that follows, whether they stay on the drug or not, if you I'm getting a little roundabout here, but it might not make sense to factor out whether someone switches or not, because that itself is the effect of the drug. So it's a simple way of estimating the effect of the drug, and I think in many cases very clinically informative. There may be other estimates that you're interested in, but as I tried to allude earlier, those estimates may be difficult to obtain. And just one more thought on this is the reference ICH E9R1 may have more to say about this. So thank you.
Thanks, Mark. Yeah, an important topic and worthwhile to think through on the front end when you're designing your analytic plan. Shabnan, question for you. With respect to the safety factor, would it be prudent to increase the safety factor if the trial is looking at a combination therapy with similar known toxicities?
Okay, yeah, thanks for that question. I think we're asking about would it be prudent to increase the safety factor for a combination product, I guess, that has similar known toxicities. That's a little tricky. We didn't actually discuss combination products, and there's actually several FDA guidances are related to the development of combination products. But I think the principles are somewhat similar, in terms of what factors warrant increasing the safety factor. With a combination product, you may have had a factorial design type of study where you would look at the efficacy and then the safety of the single products alone, administered alone, and then the safety of the combination, product. So, and that would be done in non-clinical species as well. So keeping that in mind, as I said, the factors that would warrant increasing the safety factor for a single product would probably be pretty similar for combination as well, I would guess. And, those would be, you can consider with whether any of the ones that are, listed in that guidance on estimating the maximum starting dose in first-in-human studies would apply to that combination product. So, just as an example, you could ask yourself if the toxicities may be expected to be greater, again, than the single product administered alone. You could think about whether there would be qualitative severe toxicities or damage to any particular organ system, which may warrant increasing the safety factor. Again, you would, kind of look at non-monitorable toxicities or toxicities that don't have any premonitory signs, irreversible toxicities, or even unexplained mortality as examples for warranting an increase in the safety factor.
Thank you so much. One more question for you while I have you. Is there a predetermined limit or threshold for unexpected toxicity observed during a clinical trial that would necessitate updating the investigator's brochure for an investigational drug?
Yeah, that's a good question. And I'm not sure what's meant by specific thresholds, but I think we would just have to go back to our definition of what's considered unexpected as far as an adverse event or a suspected adverse reaction. And, those are, again, if it's not listed in the investigator brochure, if it's not listed at the specificity or the severity that's been observed, if you don't have an investigational brochure, if it's not required. And then again, if it's not consistent with the risk information that's described in the general investigational plan or elsewhere in the current application. And then finally, unexpected also refers to adverse events or suspected adverse events that are mentioned in the IB as occurring with a class of drugs or anticipated from the pharmacological properties of the drug, but are not specifically mentioned as occurring with a particular drug under investigation.
Great.
I hope that answers the question. Thanks.
Yes, thank you. Mark, a couple of additional questions for you. In planning a clinical trial, we often do power calculations. Can you explain the role of power calculations in the design of clinical trials?
Okay, I'll try in the few minutes we have left. A topic I didn't have time to cover is hypothesis testing. You probably have come across it in some form or another. But typically trials are designed around hypotheses to demonstrate or that a drug has an effect. And the way that typically works is we have the alpha level, which is sort of the probability of declaring that the drug has an effect when it really doesn't. And that's where we see values like 0.05 or two-sided 0.05. So that's sort of fixed ahead of time, say 0.025. That would be the probability of declaring the drug has an effect when it really doesn't. Power is sort of the other side of that. Given that we fix that alpha and the drug does have an effect, usually that's specified based on clinical justification, what would be sort of a clinically expected or reasonable effect that this drug would have. So given that effect, we want to have the sample size such that there's good probability, usually 80 or 90%, that the trial will declare that the drug has an effect. So typically, that's how we plan sample sizes. We pick an alpha based on sort of regulatory precedent or the therapeutic precedent, and then we adjust sample size, and that's the power calculation. We adjust sample size, so given that the drug has an effect, that we have a reasonable probability of detecting that effect. So I hope that's somewhat helpful, and I think ICHD9 may have more to say about that as well.
Thanks so much, Mark, and also Shabnan for your responses today, and also for the really informative presentations. We'll go ahead and wrap this up. I'm going to hand the mic over to Leonard for some closing remarks for today. Thank you all.
Well, thanks very much, Kim, for leading the discussion, and thanks to Mark and Shabnan for great presentations. It's been a fairly intense agenda today, and we've finished with some of the very critical areas in clinical trial design and execution, which are basically safety and the statistic principles that help us interpret the data. So I also want to thank the audience for their excellent questions. Tomorrow we'll resume at 11 Eastern Time, and we'll be starting with a focus on basic science. We'll be talking about chemistry, manufacturing controls, toxicology, and clinical pharmacology, which are all very important in the investigator's brochure. And then we'll pivot to another area of great importance, which is the investigator responsibilities and inspections. And finally, we'll end with a talk on international trials. So that brings us to the end of today's agenda. And before I hand over to Ray for some administrative comments, I just want to take the opportunity to thank you all, to wish you a good evening wherever you are, and we look forward to resuming tomorrow morning. Thank you.
Thank you, Dr. Sachs. And we do have a few housekeeping note reminders. In addition to the speaker evaluation, we have our final evaluation, which is available right now from our one-stop shop link where you logged in today earlier into this event. This is the final survey, is the mechanism by which you'll receive the certificate of attendance, which will support CE credits for RAPS, OCRAS, QA, and ACRP. So be sure to download the certificate immediately upon completing the survey. Otherwise, this opportunity will be lost. Even if you do not want to read the certificate, please take a few moments to complete the conference evaluation, as your feedback will help guide the design of our future events. Note, the evaluation and certificate are only available for 2 weeks, and that takes us to the deadline of December the 21st. To summarize, for our physicians, pharmacists, and nurses claiming the continuing education credit, CEs are only available if you've registered via Adobe Connect and attend real time. SBIA will send the CE claim code and instructions to all attendees later today. So be sure and complete the CE session evaluation on or before December the 21st. I apologize in advance, but we are unable to make exceptions to that deadline. Attendees have 14 days from the last day of the activity to log in, complete the required CE evaluations, and attest to attendance to claim credit. Today's email will have the claim code and instructions together with the survey link. As a reminder, all sessions are being recorded. It'll take us a week or so to process recordings. You'll soon be able to access them from SBIA's webpage, and that is fda.gov/CEDAR SBIA Learn. On behalf of CEDAR Small Business and Industry Assistance, we hope that you enjoyed today's presentation and found them helpful. We look forward to your presence tomorrow starting at 11 a.m. Eastern Time for Day 2 of CITC 2023. Thank you for attending, and this concludes Day 1 of the FDA Clinical Investigators Training Course.